The UPSC CSE arena is an ever-evolving space where staying up to date becomes as crucial as maintaining a thorough preparation. With strategic planning and holistic coverage for exam syllabus, Vision IS Delhi presents GS Foundation course. Avail analysis and integration of current affairs with GS syllabus. All India test series with an innovative assessment system, inculcating post-test analysis, discussion, All India ranking, and more. Explore the holistic learning modules intrinsically designed for the ideal aspirant.
Good afternoon. So continuing from where we left yesterday, we discussed first two topics. In today's class, I'm going to start with biotechnology and health. These two topics, biotechnology and health. Let's start with one by one. This was a question asked by UPSC in 2016. The context of the developments in bioinformatics, that in itself is a very interesting word. Bioinformatics is that we are trying to understand how biological systems work together as one system. Think about it. A lot of things are happening in our body, how our DNA expresses into a particular messenger RNA molecule which helps in protein synthesis. So how all of that functions together. So often we use, there is an interdisciplinary branch which we call computational biology, where we apply computer science, and the goal here is to study biological processes in more detail. The term transcriptome, sometimes seen in the news, refers to. This is not a very difficult question. The only thing is that you have to know about it. But this is a very popular question, popular thing when you study biology, when you learn about nucleic acid. This is the first thing you come across. This transcription is nothing but DNA molecule which has all the information about all the proteins which can be made. But DNA does not leave the nucleus. It sends its messenger. Does not send the entire information. You think of this as a chef who knows everything what to cook, and you essentially are noting down something on piece of paper, and you are bringing that piece of paper to your kitchen to make something. So that chef who knows about everything is like DNA molecule, and that whatever you have noted it down, that can be thought of, or you can be thought of as a messenger. And then in your kitchen you can make that protein, but the information you took from that chef. So this process is called transcription, and all of the transcription together. So your DNA expresses into many type of messenger RNA molecules and helps in making hundreds and thousands of types of proteins, not just hundreds, but thousands of types of proteins. And that total set of expression together is called transcriptome, right? So that is what one of the option was, that the full range of messenger RNA molecules expressed by an organism. So for every organism, this mechanism is same. The only thing is that what type of proteins they can make or they cannot make, that will be different. But the mechanism of transcription and the second part, which by the way leads to protein synthesis translation, that will be same for all living organisms, right? So this complete set of mRNA expression of living organism, we use this word transcriptome. Why I said this is an easy question? Because this is the first thing when you study about DNA molecules, this is one of the first thing you come across, that what is the role of DNA molecule? And it performs many role. One of that very important role is this genetic expression, protein synthesis through the process of transcription and translation. So that is why I said that this is not a very difficult question. Okay. तो यहाँ पे जो आंसर है इस क्वेश्चन में वो बी है. All of these words which we are learning, in one way or another UPSC can form. So जो ये concepts है बहुत ज़्यादा concepts नहीं है, and once you understand them well, then you can answer many types of questions. There's another very famous technology we use here is called genome sequencing. Okay, and genome sequencing was in news recently also a lot. There is this Genome India project by Department of Biotechnology, where Department of Biotechnology essentially gathered genome data of many ethnicities in India, 10,000 people, and then it has completed the sequencing of all of those 10,000 sequences, and it is stored on a very powerful supercomputer. And this project was completed just last year, I would say. So genome sequencing remains an important topic. So what is genome sequencing first? This is your DNA structure. DNA structure comprises of the most important part of this structure, apart from the sugar base, which is deoxyribose, and a phosphate group, which you can see here, phosphorus. Apart from that, the most important part here is that these nitrogenous molecules, we call them base, and there are 4 of these bases: cytosine, guanine, adenine, and thymine. And these 2 DNA molecules together make a helical structure, and these base pairs are bonded together with a very weak bond called hydrogen bond. And that particular structure, that base pairing sequence, decides the properties of living organisms, like what type of traits you exhibit. Some traits which are very much visible, but other traits which may not be visible, but they still are there. So traits like your eye color, or traits like your height, for example. So many of the things are decided by by the way, I should also be very clear that some other factors also play a very important role, right? For example, if many children suffer from child stunting if they do not have adequate nutrition during young age. So it should be very clear that multiple factors decide many traits, but many of those things are essentially decided by your genetic structure. And that genetic structure का जो सबसे important part है वो आपका ये जो base pair है उनका sequence क्या है. So knowing that complete sequence together, if you write down even for one DNA molecule, you can fill an entire thick book. It's a very complex sequence. So writing that complete sequence together is called genome sequencing. And when you do genome sequencing, that allows you to know many things. For example, at its very core level, it allows us to distinguish among organisms and also find the similarity within one species. Jaise, we all are 99.9% same, or 99.8% at least same. There is a 0.1-0.2% difference, and that 0.1-0.2% difference is how all of us are unique. But we are also about 98% similar to chimpanzees, our closest evolutionary ancestor. But that 2% difference makes a lot of difference. And then chimpanzees and human beings ka jo difference hai, that's very visible. So it allows us to understand the relationship among organisms. It allows us to understand how evolution has occurred. It allows us to understand about how some people can be predisposed to certain type of diseases. There are people who have never smoked, drank alcohol, and still suffer from lung cancer or liver cancer, right? Why certain people are more predisposed to, let's say, diabetes. So all of these questions can be answered. And of course, it can also answer about genetic disorders itself. So genetic predisposition as well as genetic disorder can be understood much better if we can do this genome sequencing, where we know the complete sequence of the base pair. There are many projects done. There was a project in 1990 which did the complete genome sequence of human being for the first time, but it took a lot of time, more than 10 years. As of now जो genome sequencing techniques हैं वो इतने advanced हैं कि कुछ घंटे में एक पूरा complete genome sequencing कर सकते हैं. So in any case, this question is about genome sequencing in agriculture. With reference to agriculture in India, how can the technique of genome sequencing often seen in the news be used in immediate future? It can be used to identify genetic markers for diseases, resistance and drought tolerance in various crop plants. The answer is yes. All of these properties are genetic traits, and genome sequencing allows us to understand that. The techniques help in reducing the time required to develop new varieties of crop plants. That is also possible. It can be used to decipher the host pathogen relationship in crops. That is also possible. So all three of these things are possible with help of genome sequencing. Answer here is D, 1, 2 and 3. You just have to remember that the purpose of genome sequencing is to understand that sequence of base pair. And that sequence of base pair exactly what makes us all of unique. And I'm not including just human beings, I'm including all living organisms. So living organisms, the properties they exhibit, how do they look like, what type of proteins they make, what are their life functions, all of those things are essentially decided by their genetic structure, and genome sequencing allows us to understand that. So the answer here is all three of them, which by the way is also true in case of जो human genome project, जो अभी मैंने example लिया human genome project का. One of the objective of this human genome project is that apart from of course its applications in healthcare and medicine development, one of the application also is that how as Indians we are related to each other, right? So we already are finding some really interesting points. I came across a very interesting article few months ago that how a group of people in Kerala, they trace their origin from northwest frontier in Punjab, right? So they migrated to Kerala region about 700 800 years ago. I'm forgetting that name of the group of people, warrior class of people. So you come across many such interesting studies, how Hadappans are related to North Indians or South Indians, things like that, all of those things. So there are applications in anthropological studies, in historical studies, in understanding how populations are related to each other, right? So genome sequencing has many applications, not limited to healthcare and medicine development. But of course that is the most prominent application. तो यहाँ पे जो आंसर है वो D है. Right now when we do genome sequencing, then DNA is divided into many small fragments, and all of those small fragments are sequenced together, and then with help of specialized softwares, the proper sequencing arrangement is done at the end, and that has escalated the process of sequencing. What used to take years and then months, now it happens in few hours. देखो यूपीएससी के क्वेश्चन कितने स्ट्रेट फॉरवर्ड होते हैं. दे आर नॉट इवन गोइंग इनटू सम टेक्निकलिटी ऑफ थिंग्स. They are simply asking about that, do you know what this term is all about? And the best part is that ऐसा भी नहीं कि ये term कोई randomly कहीं से भी pick कर लिया है. ये जो कुछ biotechnological techniques हैं, they often remain in news. That somatic cell nuclear transfer technology remains in news, and that is nothing but reproductive cloning of animals. And I can explain this with help of this diagram, that in somatic cell nuclear transfer, we take a somatic cell are these diploid cells in your body, basically जो भी non-sexual cells हैं, जहाँ पे भी आपके 23 pair of chromosomes हैं, they are essentially somatic cells. Compared to जो non-somatic cells हैं, basically they are sexual cells. जो sperm और egg है, these are non-somatic sexual cells. They only have 23 chromosomes, not 23 pair, but only 23 chromosomes. So in somatic cells, so you take a somatic cell, you remove the nucleus, and then you take an egg cell and you remove the nucleus from the egg cell. And in this egg, you insert this nucleus. So now I have an egg cell which has nucleus, which means the complete set of DNA here. And now we can replicate this and make into clone. We can use this clone जैसे to make an animal also. It was used for the first time for a sheep. आपने पढ़ा होगा ये चीज़. What was the name of that sheep? Dolly sheep, yes, exactly. Dolly died recently. I remember this reading this news. But it can also use to for therapeutic cloning, that you are trying to make some sort of protein or some tissue which can have applications in medical sector. So that is also possible. But this technology, अब नाम दोबारा से देखो, that somatic cell nuclear transfer technology, and you get the idea that essentially we are talking about cloning of animals. So answer here is C. And you'll find many such questions where only the term is mentioned, and you're supposed to simply know that what that term is all about. There are many other examples in last few years. In fact, I can show you perhaps one more question very similar to that. See a question देखो. In 2020, in the context of recent advances in human reproductive technology, pro-nuclear transfer is used for. अब यहाँ पे जो आंसर है वो D है. अब ये specific information पता होना चाहिए. That prevention of mitochondrial diseases in offsprings, that is the correct answer. Why that is correct answer? So, and why this was asked? To appreciate that, you should know something called 3-parent baby, जो बहुत ज़्यादा न्यूज़ में है. हमारा जो DNA है, if you take any of our cell, then the most important organelle is nucleus. Inside nucleus, I have this nuclear DNA. But turns out that we also have a very small DNA in mitochondria, which does not have a lot of genes. It has hardly like few genes, 20 of them, I don't know, or 17 of them, something like that. It can make some type of proteins. So we call this mitochondrial DNA. And mitochondrial DNA we only inherit from our mother, right? So my mitochondrial DNA, it's same as my sister's mitochondrial DNA, and both of us have only inherited from our mother. Mitochondrial DNA खुद में एक बहुत ही amazing चीज़ है कि ये कहाँ से अचानक से आ गया. ये mitochondrial DNA कहाँ से आ गया. न्यूक्लियर DNA तो चलो ठीक है, समझ में आता है. ये अलग से पूरा माइट्रोकॉन्ड्रिया है क्या? दिस सो कॉल्ड पावर हाउस ऑफ सेल हम बोलते हैं ना माइटोकॉन्ड्रिया को. एंड जो इसका सबसे वेल एस्टैब्लिश्ड थ्योरी है, तो ये कहाँ से आया है? ऑल्दो दिस थ्योरी we do not have like evidence to confirm it, we simply cannot confirm it, but the best theory is that perhaps billions of years ago some bacteria was infected with some other microscopic organism, maybe some archaea or some virus, as a parasite. But in the course of evolution, they became a symbiotic organism. And whatever that parasite or whatever that archaea was, it essentially led to a conjugate organism. And that particular, what was a parasite, evolved into mitochondria. And because of that, you have this entirely different set of mitochondrial DNA, different from nuclear DNA. So anyways, that is another, I digressed a little bit. We already know about genetic disorders. We know that some genes are not functioning properly, which can lead to various type of problems, right? Sickle cell anemia, thalassemia, so many diseases are there, which are genetic disorders. But there are also genetic disorders which occur because of mitochondrial DNA defect. So I can have nuclear DNA defect disorders, but I can also have some diseases which occur because of if there are defects in mitochondrial DNA. And mitochondrial genetic defect को treat करना ऐसे भी मुश्किल है बहुत. आप symptoms को manage कर सकते हो, but mitochondrial DNA defect को treat करना और मुश्किल है. तो एक जो technology which has been developed, which we call 3-parent baby, that we know that mitochondrial DNA you only inherit from mother. So if you know that there is a woman and there are some the mitochondrial DNA is defective, there are some genetic disorders in mitochondria, then when this woman marries a person, then for the baby, we can have some sort of setup where the mitochondrial DNA will be replaced with a healthy donor, right? So now this baby still has the nuclear DNA from biological father, biological mother, but there is a donor here, a woman. Remember this 3-parent baby, 2 mother, 1 father. So there is a donor here, and that donor has healthy mitochondria, and that healthy mitochondria is replacing that defective mitochondria. And there are 2 ways to do that. One of that method is called pro-nuclear transfer, and another of the method is called maternal spindle transfer. In maternal spindle transfer, for example, we take mother's egg, the sexual cell, unhealthy mitochondria, and we take donor's egg, which has healthy mitochondria. We replace the nucleus from mother's egg, and we replace the nucleus from donor's egg also. But in the donor egg, now I have healthy mitochondria, and this nucleus from mother, biological mother, is inserted inside this egg cell. So now I have an egg cell which has healthy nucleus, which has healthy mitochondria, and now fertilization with sperm can happen, and the baby will not suffer from mitochondrial defect. So this is maternal spindle transfer. But there is one more way to do that, which is called pronuclear transfer. That same thing, unhealthy mitochondria. So fertilization happens to both of these eggs. So we fertilize both of these eggs, donor egg also and mother's egg also. And that fertilization leads to growth of what we call pronuclei. And then we remove the nucleus, and we remove the nucleus here also, and we replace this pronucleus, which is healthy, into this pronuclei, which has healthy mitochondria. So in this case, the only thing is that fertilization is happening, that mitochondrial replacement is happening in this case before fertilization, and in this case it is happening after fertilization. Both of these things are possible, and UK became the first country to legalize it. They already have few babies which have been born with this 3-parent baby concept, and in this context this topic was asked, and the answer here is D, prevention of mitochondrial diseases. And immediately next year there was another question on this topic. (2021). In the context of hereditary diseases, consider the following statements. Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy, either before or after in vitro fertilization of egg. That is correct. And a child inherits mitochondrial diseases entirely from mother and not from father. That is also correct. Answer is both 1 and 2. So you find that how from one technology or one theme, many type of questions can be asked. कल के क्लास में भी हमने देखा था, अभी भी examples हैं, that at least we found few questions from this topic. Anyways, let's go back to this was the question. GM mustard, ये बहुत बार तब भी न्यूज़ में था. In 2017 it was in news a lot, and it is again in news a lot in last 1-2 years. So I will not be surprised if UPSC repeats this theme again, either in prelims or mains, or at least in mains they can pick this theme of GM crop in general without going into this DMH11. So this GM mustard is called DMH11. GM mustard has the genes of a soil bacterium that give the plant the property of pest resistance to a wide variety of pests. देखो ये एक बहुत ही specific, बड़ा ही interesting statement है जो कि गलत है. बट ये अगर आपने डिटेल में नहीं आपने पढ़ा है तो इसको आपको लगेगा कि हाँ ठीक है, बहुत सारे GM क्रॉप का एक दो बेनिफिट यही होता है ना, that we want to develop this pest resistance. So you may think that this statement is correct, but this statement is wrong. I will come to why it is wrong later. GM mustard has the genes that allow the plant cross-pollination and hybridization. That is the actual purpose of GM mustard, and that is why first is wrong. Third option was a factual statement, but this was a very popular fact. I remember reading a lot about GM mustard, and it was very clear that this is made by Indian scientists. तो सबसे एक अच्छी चीज़ ये थी that we are not dependent upon some foreign multinational company. तो जो farmers exploitation की हम बात करते हैं वो वाला issue यहाँ पे नहीं था. But it was not made by Punjab Agricultural University. It was made by Delhi University, scientists in Delhi University, and not by IARI. I think Pusa Agriculture Institute was involved into that. So third statement was wrong. If you eliminate third, you have your answer 2 only. Right? Third is a factual statement, but if you know that fact, that this is wrong, then you know that first is also wrong. Now why first is wrong? Mustard, most of the mustard varieties are self-pollinating. Right? Mustard is a type of plant which is self-pollinating, which essentially mean that desirable traits cannot be passed on to the future generation. So the purpose of this gene modification two specific enzymes are being added here. If I'm not mistaken, the names are Barnase and what is the second name? If anybody remembers. So two very specific enzymes are being used here, recovered from soil bacteria. That part is correct actually. But the purpose here is not pest resistance. The purpose is that let's make this part possible. Let's make that cross pollination and hybridization possible, which is not possible for the traditional varieties of mustard. The traditional varieties, they are self-pollinating. And because of, let's say, if soil condition has worsened, climate has changed, and if plants are self-pollinating, it means that they are making an identical genetic copy, which means that if the traits are not desirable given the how climate is changing or how other things are changing, then mustard yield is going to reduce gradually, and it can become extinct also. At its very worst, it can become extinct also. Not immediately, but maybe in hundreds and thousands of years. So that is where this cross pollination and hybridization, which has been made possible with help of genetic modification, and we are calling this variety DMH11. So that is why first statement is wrong. The main purpose was second, and third is also wrong. Answer here is 2 only. ये दोबारा से न्यूज़ में इसलिए था because in India the institution responsible for doing field trials and ascertaining that if a particular GM crop is safe or not is Genetic Engineering Appraisal Committee under Ministry of Environment. And they had given this approval in 2017 also that it is safe. And they repeated this, they reiterated this in 2023 that it is safe. But even then, Government of India, there is a lot of pressure that we should not introduce GM crop. It can lead to genetic pollution, a number of threats, possible threats. And because of that, Government of India has not actually approved this. This is another very interesting question. With reference to the recent developments in science, which one of the following statement is not correct? Right. So out of 4, 3 are correct. ऐसे question I always feel that you should attempt. और अगर correct भी पूछ रखा है कि which of the following is correct, and ABCD में पूछ रखा है, even then you should attempt. You'll find that there are many options to eliminate, like wrong options वो आप highlight हो जाएगा. तो यहाँ पे आपको ये चारों में बस ये देखना है कि चारों में कौन एक सबसे ज़्यादा मुश्किल है अचीव करना. जैसे एज एस्पिरेंट अगर मुझे नहीं भी पता होता तो इतना तो मेरे पास एप्टीट्यूड है कि मैं इन चारों में ये डिसाइड कर पाऊँ कि विच ऑफ द 4 इज द मोस्ट डिफिकल्ट थिंग टू अचीव इन अ लैब सेटअप. Functional chromosomes can be created by joining segments of DNA taken from cells of a different species. Okay, now there are two parts of this statement. The first part is functional chromosomes can be created. And the second part is by joining segments of DNA taken from cells of different species. This part has been done like few decades ago. We call this recombinant DNA. So recombinant DNA technology is not a new thing. This was done like many, many years ago. But saying that you have made a functional chromosome, because the word chromosome means a lot of things. Chromosome, DNA is a very complex, lengthy molecule. In your cell, you have this chromosome where you can think of this as एक ऐसे समझ लो कि एक सिंपल सा नीडल है और उस नीडल के टॉप पे पूरा DNA molecule wrapped है with help of some proteins. This complete structure together we call it chromosome. So saying that you can create a functional chromosome, we are not anywhere near that. We can create recombinant DNA. This thing has been done like few decades ago. In fact, even before gene editing, we were able to develop recombinant DNA. But that functional chromosome part is not true. So यहाँ से भी answer आ गया. But let's look at the other option. Pieces of artificial functional DNA can be created in lab. This has been again made possible. If it is only about artificial, because remember DNA is a molecule. Can we make a molecule into lab? The answer is yes. So this has been done. A piece of DNA taken out from an animal cell can be made to replicate outside a living cell in a lab. This has been done again many years. And जो last वाला है वो तो सबसे simple है. Cells taken out from plants and animals can be made to undergo cell division in lab petri dishes. C and D should have been very clear that ये तो नहीं होना चाहिए. Some confusion can occur between A and B, but if I have to choose between A and B, I will choose A to be not correct, because that statement that you have made a functional chromosome, because remember, chromosome is this very tightly coiled structure which carries your DNA molecule in a very small space, because if you untangle the entire DNA, then it is going to take a lot of length, but all of that length of few meter is essentially on this needle volume. It is so tightly packed inside our cell. So saying that we have done this, that does not make sense. So that is why जो A है वो गलत statement है यहाँ पे. Then there is this another very interesting technology called RNA interference. You can appreciate RNA interference even more with this new Nobel Prize in medicine given for microRNA, discovery of microRNA. There are many type of RNA molecules. In previous question we talked about messenger RNA. But protein is actually made in ribosome. So turns out ribosome has its own RNA which is called ribosomal RNA. This messenger RNA transfers this information to ribosomal RNA with help of something called transfer RNA. Then there are another interesting example of RNA molecules called silencing RNA and microRNA. So in almost all eukaryotic organisms, so except bacteria, almost all eukaryotic organisms, including human beings, there is a natural mechanism where certain genetic expressions are restricted. That a messenger RNA, which ideally could have led to a translation of a protein, but our body has this mechanism that if this protein is not desirable, if it is not required, then we need to stop the process. Right? So go back to that example that आपने शेफ से डिश ले लिया, लिख लिया पेपर पे, आप किचन में गए, बट आपने देखा कि ये जो है ये तो नॉनवेज डिश है और आपके घर में तो नॉनवेज बनता नहीं है, तो कोई आपको स्टॉप कर रहा है कि नहीं ये नहीं बन सकता यहाँ पे. सो यू कैन थिंक ऑफ दिस एज RNA interference. That RNA interference is a natural mechanism which occurs in all eukaryotes, not just in mammals, all eukaryotes, including human beings, that certain genetic expressions do not come to fruition. And there is a natural mechanism for that. And that also occurs because of some other RNA molecules, such as silencing RNA or microRNA molecules. And in that context, this question is asking. It is used in developing gene silencing therapies. So once we understood that this is happening naturally, that there are certain specific RNA molecule which can disrupt some genetic expression, then we ask this question that can we do this in a lab setup? Like, can we make such RNA molecule and then can we silence some genes? Maybe silencing those genes is desirable in plants or in animals or even in human beings for that matter. So first is true that yes, it can be done. It can be used in developing therapies for the treatment of cancer. That gene silencing का एक जो सीधे direct application है, वो cancer treatment है. That some genetic expression which was not desirable, but since it is happening, it is producing some mutated growth, some mutation is occurring in your body. So silencing those genes with help of silencing RNA molecule can actually allow us to find solution at the very root cause of the problem rather than again tackling symptoms. So even second is true, and fourth is also true. It can be used to produce crop plants that are resistant to viral pathogens. That virus attaches itself to host DNA, right? So again, figuring that out, that how to take care of that, how can we silence that part, we can do this also. So if I know 1, 2 and 4 to be correct, then my answer is clear. But why third is not correct? It can be used to develop hormone replacement therapy. अब देखो, hormone replacement कुछ नया नहीं है. Hormone replacement बहुत सालों से चल रहा है. Hormone replacement therapy के लिए हमको इतने advanced method की ज़रूरत ही नहीं है. जैसे मान लो कि एक simple सा hormone replacement therapy is कि जो insulin जो लोग लेते हैं जो diabetes से suffer कर रहे हैं. That's a very simple example of hormone replacement therapy. तो इसके लिए हमको किसी RNA इंटरफेरेंस, किसी जीन मॉडिफिकेशन, इन चीज़ों की ज़रूरत नहीं है. Yes, some glands not producing enough hormone, right? And we can essentially invest into hormone replacement therapy. That can be done. You ask a question that can we use RNA interference to silence some genes, which essentially is helping in hormone production. So first of all, it is silencing, not replacement. But that answer is yes, we can do that. But the point is that we do not have to do that. We do not have to go into such complicated thing for something which we figured out many years before this entire RNA interference technology. So we are not using this RNA AI for hormone replacement therapy. And that is why the third statement is not correct. But I was really glad to see that UPC also did not give this option 1, 2, 3 and 4 just to give you cause this extra confusion that if 3 is correct or not. If I know 1 and 2 to be correct, even then I have my answer. Answer is 1, 2 and 4. Okay. What is Cas9 protein that is often mentioned in the news? I talked about that in the last class. That in 21st century, one of the most remarkable discovery has been this mechanism called CRISPR-Cas9. Two group of scientists, Jennifer Doudna, Emmanuel Charpentier, they are no less than some of the greatest scientists of all time. This two group of scientists and their team, they were investigating the immune system of bacteria like, let's say, E. coli. And they found that in some bacteria there is this immune system. So remember, viruses can infect bacteria also. कल के क्लास में हमने डिस्कस किया था, एक जो बेसिक क्वेश्चन ऑफ बायोलॉजी था. एंड जिस तरीके से हमारे बॉडी का एक इम्यून सिस्टम है, इफ वी आर इन्फेक्टेड बाय पैथोजेंस, our immune system gets activated to tackle this. Even bacteria has an immune system that gets activated to tackle viral infection. In some bacteria, in not all of them, in some bacteria, the mechanism is, we have given it a name, CRISPR-Cas9. There is a very good reason why we call it CRISPR. Uska khud ka full form hai. So how this mechanism works is that bacteria ka ek pura part of DNA hai which carries the information about all of the viruses which it has been infected in the past. And then it keeps on, if there is, let's say, a viral infection, then it's like a ticking time bomb. Bacteria has few minutes or few hours before this virus completely hijacks the bacterial DNA and starts multiplying using bacterial DNA. So the moment it happens, the bacterial immune system kicks in, and it essentially leads to this protein called Cas9. And it keeps on, now it has a like starting point, end point, and it keeps on looking for the viral DNA. It attaches to one segment of DNA, sees that if there is a viral DNA or not. If there is not, goes to the next segment. And the moment it finds the correct viral DNA, it produces this protein called Cas9, which acts as a molecular scissor. And it removes that particular viral DNA part. So this process happens naturally, and then bacteria remembers this viral structure. This process happens naturally in many bacteria. So this group of scientists asked this question, that can we replicate this into lab? Can we also make a guided RNA molecule to look for a specific segment of DNA? And then can we make this molecular scissor Cas9 to remove that specific DNA? Can we do that? And years of research led to the conclusion and a result that we can do that, and they did that, and they were given Nobel Prize in chemistry, I guess 4 years ago. In 2020, these two scientists received Nobel Prize in chemistry for this. So in this question, UPSC did not even ask about some detailed mechanism of CRISPR-Cas9, although I will not be surprised that in mains somewhere down the line there will be a question about what is CRISPR-Cas9, what are its applications, things like that. And if there is a question in mains, ideally you should write some major themes we discussed. So यहाँ पे आंसर बहुत सिंपल है, molecular scissors used in targeted gene editing. पहला ही स्टेटमेंट जो है करेक्ट है, बाकी हमको देखने की जरूरत भी नहीं है. Another question on advancements in biotechnology, that genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. This is called germline editing. And CRISPR-Cas9 is so accurate and so precise that yes, it is possible. With older techniques, it was not that much possible. But CRISPR-Cas9 can actually allow you to do that. And this topic, by the way, was in news for a very interesting reason. In 2020 itself, or I think 2019 or 18, a Chinese scientist announced that he has done this germline editing to a zygote, which will not be infected by HIV. Now, you prima facie, you may think that this is such a great news that we are able to do this gene editing, germline editing, and now the baby cannot be infected by HIV. But this is a slippery slope. One of the biggest criticism of gene editing has been that if people start selecting desirable genes simply because they can, right? And there is this threat of what we call designer baby, that rather than letting natural selection do its work, you are selecting which genes you want. You want a baby with blonde hair and blue eyes and so on and so forth, right? We do not want this is not how evolution should function. So this has been always a criticism of gene editing, and I remember very specifically that when this was announced, of course it led to a global hue and cry that this is a slippery slope. It can lead to this entire concept of designer baby. The scientists were shunned, this work was stopped, and Jennifer Doudna in fact came out publicly and asked everybody that we need to take a pause on use of CRISPR-Cas9, and we need to first think about how should it be used, for what purpose should it be used. Like, so the person, she herself, like who discovered CRISPR-Cas9, it's saying that we need to take a pause here. So, but the point is that this is correct. And this is, however, illegal in many countries, including in India. जो germline editing है, that editing germ cells, sexual cells, is banned. And even this is true. This is also part of germline editing. Person's genome can be edited before birth at early embryonic stage. This is also part of germline editing. This is also banned, but yes, both of them are possible. Human-induced pluripotent stem cells can be injected in the embryo of a pig, and vice versa is also true. So we are doing this already to see that how stem cells of different organisms, because हमारा जो anatomy है वो pig के anatomy से बहुत similar है. So बहुत बार इसीलिए आपने देखा होगा कि जो हम बोलते हैं ना ये organ transplantation में हम pigs को use करते हैं, उसका बहुत बड़ा कारण जो similarity in anatomy है. So we are seeing that how it affects pigs, and if somehow we can suppress the reaction, then the next step will be now doing this in the opposite direction. And this will have again a lot of therapeutic applications. So answer here is 1, 2 and 3, that all three of them are possible, right? But first two are illegal and third requires huge regulation. The third है वो ऐसे कोई भी नहीं कर सकता, इसके लिए भी बहुत सारा rules regulations होंगे. हर institute will invest into a lot of rules and regulations. Only then it can be done. This for me is a question which is difficult, but is attemptable given the options. Even if you do not know a lot about this question, you should be able to, you should try to attempt. I always feel that अगर option ये नया वाला pattern नहीं है, how many of these statements are correct? और 4 statement या 5 statement का question है. Statement का, एक होता है statement, एक होता है खाली 2 वर्ड का दे रखा है 4-5. आई एम नॉट टॉकिंग अबाउट दैट. जैसे आशियान में कौन कौन से कंट्रीज हैं, आई एम नॉट टॉकिंग अबाउट दोज क्वेश्चंस. आई एम टॉकिंग अबाउट जो प्रॉपर स्टेटमेंट बेस्ड क्वेश्चंस हैं. आई ऑलवेज फील लाइक दैट यू शुड ट्राई टू अटेम्प्ट देम, बिकॉज यू विल हैव मेनी options to eliminate and arrive at the right answer. जैसे if I look at this option, then ये तो बहुत clear है कि कम से कम एक statement wrong है. There is no option called 1, 2, 3 and 4, but as many as even 3 statements can be wrong. The moment I know that, I should first look for right statement. Believe me, looking for right statements is simpler compared to looking for difficult statement. So अगर मुझे ये पता है कि इसमें सब सही नहीं है, then I should first look for the most correct option, the most right option. जैसे a substantial saving in seed material is made when bud chip settings are raised in a nursery and transplanted in the main field. When direct planting of sets is done, the germination percentage is better with single budded sets as compared to sets with many buds. If bad weather conditions prevail when sets are directly planted, single budded sets have better survival as compared to large sets. In that second and third, you find a commonality that it is trying to compare multi-bud versus single bud. And in both the second and third statement, it is saying that single bud is better than multi-bud. So one logic which you can think of is that maybe these two statements are either correct together or either false together. I do not know, right? Sugarcane can be cultivated using settlings prepared from tissue culture. ये आपको पता होना चाहिए. ये सबसे simplest statement है that ये tissue plant tissue culture जो है बहुत पुरानी चीज़ है. Again ये petri बहुत सालों से लोग कर रहे हैं. एंड शुगर केन का देखो होता है प्रॉब्लम क्या होता है कि जब आप कल्टिवेट करते हो सेम क्रॉप से करते हो, बट देन कैरिंग दिस, ट्रांसपोर्टिंग दिस, स्टोरिंग दिस, ऑल ऑफ दिस बिकम्स वेरी heavy. There is a huge logistics cost. So rather than doing this, what we call is bud chip setting, that we take buds which is more than enough for this sugar to cultivate, and in a specialized setup then we grow them, which may look something like this. We take these buds, we grow them and then we transplant them. तो इसका बेनिफिट ये है कि जो हमारा पूरा लॉजिस्टिक्स कॉस्ट वगैरह है वो हमारा कम हो जाएगा, जो ट्रांसपोर्टेशन कॉस्ट है वो हमारा कम हो जाएगा. सो दैट इज वाई फर्स्ट स्टेटमेंट इज ट्रू एंड फोर्थ स्टेटमेंट इज ट्रू. इफ आई नो दैट, then I know that answer will be 1 and 4 only, and which tells me that in both second and third the exact opposite is true. Which, by the way, if you know about buds, so if there are more such buds, single buds versus multiple buds, 2-3 buds आपको दिख रहे हैं अलग अलग, तो अगर एक सब possible है कि all of these buds are, there is a possibility that they can grow into a larger sugarcane plant. So in case of adverse condition, if single bud does not survive, then you cannot do anything. But in multiple buds, the chances of survival is higher because all of the buds can grow into a plant. So that is why second is not correct, third is not correct, the exact opposite is correct. First is correct, fourth is correct, answer is C here. I repeat, I totally acknowledge that this is a difficult question, but I do feel that if you spend some time, and even if I am coming across this theme for the first time, and I also know that I'm doing this analysis post facto outside examination hall, while doing the same analysis during examination is much more difficult. I also accept that, I acknowledge that, but at the same time, I feel like that these are those questions which are not factual, some logic is involved, some analysis is involved, and if you spend some time, there is a good chance that you'll get such questions wrong compared to factual questions. Vaccines पे question expected था, prelims में भी आया था, mains में भी आया था because of COVID-19. But they did not ask about mRNA, they did not ask about anything. They asked about altogether a different type of vaccine, recombinant vector vaccines. Consider the following statement. तो वैक्सीन बनाने का देखो मेनली 3 स्ट्रैटेजी है. उन 3 स्ट्रैटेजीज में अलग अलग टाइप के वैक्सीन्स है, बट स्ट्रैटेजी 3 टाइप का है. इन फर्स्ट वी टेक द इंटायर पैथोजन, जो भी पैथोजन है, बैक्टीरिया, वायरस, वी टेक द इंटायर पैथोजन ईदर इन इट्स वीकेंड फॉर्म और इन इट्स डेड फॉर्म, and then that vaccine is injected or orally provided. So that is called live attenuated vaccine or inactivated vaccine. ये मेरा पहला approach है. That when it is injected, body recognizes this as an antibody, recognizes it as an antigen, produces an antibody against it, and when you actually are infected, body can neutralize that. Second approach is what we call subunit approach. In subunit approach, I do not have to take the entire pathogen. I need to recognize that in this pathogen, there is this antigen part. Either it is part of the pathogen or it can also be some protein that pathogen is producing. So I make my vaccine not using the entire pathogen, but only the part which is more than enough to elicit an immune response in the body. That is called subunit approach. And recombinant vector vaccine is example of subunit approach. And then third approach is of course this nucleic acid approach where you have messenger RNA vaccine or DNA vaccine. So when we are using that subunit approach, using part of pathogen or some protein, then yes, we can further do genetic engineering in development of these vaccines so that we will try to ensure that body can elicit a more stronger response. So that part is true. And bacteria and viruses are used as vectors. So हम क्या करेंगे, हमको जब vectors का use यहाँ पे दो तरीके से है. That in जो भी हमने ये कोई मान लो कोई प्रोटीन हमको बनाना है, so हम क्या करेंगे, we want this to be occurred at a very rapid rate. तो हम क्या कर सकते हैं कि हम वायरस का यूज़ कर सकते हैं. वायरस को हम पहले उसका जो भी इन्फेक्टिविटी पार्ट है that needs to be neutralized, and we can modify it, we can use it as a vector, as a carrier of this particular thing. And then as this virus multiplies, we have many copies of this protein, and I can make a vaccine based on that. So even second is true, and not we can also use bacteria for that. So first statement is correct, second statement is correct. In 2021, there was not a question on mRNA vaccine or COVID vaccine, but there was a question on vaccine. Here the answer is C. But immediately in 2022, there was now a question on COVID vaccine. I do not expect such questions, at least this factual questions I do not expect anymore. ये current affairs inspired था. Serum Institute of India produced COVID-19 vaccine named Covishield using messenger RNA platform. वो बिल्कुल ही गलत है. जो mRNA vaccine, जो जो Covishield vaccine था, that essentially was a live attenuated vaccine. And by the way, that did not even use coronavirus. That used adenovirus. It made some modification to adenovirus. So that adenovirus का भी थोड़ा spike होता है, but that spikes were changed with the spikes of coronavirus. Sputnik 5 is manufactured using vector-based platform. That is true. Covaxin is an inactivated pathogen-based vaccine. This was indigenous vaccine, and yes, it was inactivated. That is true. If I know first to be wrong, my answer is 2 and 3. But yes, the first two vaccines which were approved in the world, not in India, in the world, in America particularly, for the first time, for any diseases, messenger RNA vaccine was approved. First was made by Pfizer and second was made by Moderna. And both of these companies had made a mRNA based vaccine to tackle the problem of COVID-19. But Covishield was not a mRNA vaccine. So here answer is 2 and 3 only. ये अब देखो, ये कोई करंट अफेयर्स एज सच है नहीं, बस आपको खाली ये पता होना चाहिए था कि इंडिया में जो इकलौता जेनेटिकली मॉडिफाइड क्रॉप लीगली अलाउड है वो आपका जीएम कॉटन है, बीटी कॉटन जो है. एंड जो ट्रेडिशनल कॉटन वेराइटी था दैट सफर्ड फ्रॉम दिस बॉलवर्म अटैक, अ पिंक बॉलवर्म अटैक. सो a genetic editing, gene editing technique was used using a soil bacterium to provide this pest resistance against this particular bollworm attack. And rather than using this technicality of what bollworm, what technique, it was simply called ball guard. And ball guard was further improved. That technique was called ball guard. It was further improved. So that ball guard 1 and ball guard 2 are nothing but genetically modified varieties of BT cotton, right? So answer is B here. And even today, if you go on Amazon and if you go on many e-commerce websites, you can buy this ball guard cotton seed. That essentially is a genetically modified variety of cotton. Ball word here is from this bollworm attack, pink bollworm attack, which used to cause a lot of problem to cotton in India. This was a question inspired from current affairs. Biofilms. Biofilms हमने देखा है बट हमको पता नहीं होता है कि ये बायोफिल्म है. अब बहुत सारे सरफेसेस देखोगे कि कुछ तो एक स्टिकी सा सब्सटेंस हो रहा है, कलर डिसकलर हो रहा है. दिस कैन हैपन इन सम केसेस टीथ ऑल्सो. It can happen on implants also. So what happens is that a particular place, when it is invaded by bacteria, and if the conditions are optimum, then they also secrete this enzyme outside their body. So it is this extracellular growth. It is not growing inside their body. They secrete this enzyme. You can think of this as a protective layer, that they create some sort of fort around them. And inside this protective layer, now they can grow really, really nicely. And this protective layer is going to give them protection from outside changes in the environment. So if a surface is not being cleaned and has been invaded by some bacteria, then that surface can lead to growth of biofilms. And since inside this film they have protection, they can multiply rapidly, and as bacteria multiply, they can also develop a strain, and that strain can be antimicrobial strain. And that was the question asked by UPSC. The biofilms can form on medical implants with human tissues. This is true. Biofilms can form on food and food processing surface. This is true. Biofilms can exhibit antibiotic resistance. This is also true. So all three of these statements are true. And this question was directly picked by UPSC from current affairs. तो ऐसा भी नहीं है कि कुछ random कुछ भी उन्होंने theme उठा लिया. They used this from current affairs and they made, in fact, the news was that how, if you in fact article अगर नहीं भी जाओ तो आप यहाँ पे news का heading भी पढ़ लो, that microplastics in sewage become hub for drug resistant bacteria, and then आप पढ़ोगे, then it talks about that how these microplastics are providing more and more biofilms where bacteria can multiply much more rapidly. So the answer here is 1, 2 and 3. Okay, what is DNA barcoding? In DNA barcoding, what we do here is, देखो अगर हमको किसी species का समझना है कि वो उसका genetic structure, based on genetic structure किस type का species है, तो हमको पूरे DNA की भी ज़रूरत नहीं है. We only need to take like few base pairs, only up to even 5, 10 base pairs are often more than enough to determine what type of species it is. So what generally is done by institutes, which can be government institutes or research institutions in general or even private companies, that they create a database of DNA of species. And if, let's say, for example, they have received some sample from wild, from forest, any DNA sample, then they simply take that small fragment of DNA and they match this with their database, that if this matches with the database, then they immediately know that what type of species it is, और ये DNA कौन से species का है. तो ये जो पूरा technology है, this is called DNA barcoding. Very simple. That we are taking some sample DNA and we are matching that sample DNA with help of our DNA, like we have some sort of portal where we have a lot of information, and then we are seeing that if there is a match, then we have identified that ये कौन से species का DNA है. So now this question is, look at this question. Distinguish among species that look alike. That's very much possible with help of DNA barcoding. Think of tiger and lion, they look alike. Or think of a leopard and a cheetah, they look alike, but clearly there are genetic differences, and DNA barcoding can allow us to distinguish that. Identify undesirable animal or plant materials in processed food. Yes, if you're eating some food, if you feel that it is genetically polluted, then bring that food to a lab. It will try to recover, if there is some DNA sample, it will recover that, will match that if that DNA sample is actually some undesirable plant or animal which is not required in the food, we can do that. But first we cannot do. Assessing the age of plant or an animal, that cannot be done with help of DNA barcoding. Right. So ये जो first part है, ये हम कर सकते हैं, जीनोम स्टडी करके आप एज एक्चुअली डिटरमिन कर सकते हैं, पर थोड़ा ज़्यादा डिटेल लेवल ऑफ स्टडी करना पड़ेगा. उसके लिए और भी चीज़ें आपको इंक्लूड करनी पड़ेंगी. सेलुलर ग्रोथ रेट क्या है, टिश्यू किस तरीके से रिकवर करता है, बोन डेंसिटी कैसा है. सो मल्टीपल फैक्टर्स आर यूज्ड एंड आई एम टॉकिंग अबाउट इन फैक्ट इन केस ऑफ ह्यूमन बींग्स. But DNA barcoding cannot assess the age of plant or an animal. It only can tell you that which plant it is, and it can distinguish between plants, it can distinguish between animals, but it cannot tell you the age of plant or animal. So answer here is 2 and 3. Answer is D here. But even when I see this question also, I know that at least one of these statement is wrong. Like statement options dekho, 1 only, 3 only, 1 and 2, 2 and 3. So I know that at least one is wrong and at least one is correct. इतना तो clear है ना मुझे? तो same logic जो मैंने अभी पहले वाले question में लगाया था, I will use the same logic. I will try to find at least one correct statement out of this 3. Even though I do not know the technology DNA barcoding, I know the function of DNA. Then I can clearly say that that second should be true. DNA का एक जो, because हर species का अपना DNA अलग है, then that second should be true. So I should find options with 2. That leads to C or D. Now I only have to worry about that if 1 is correct or if 3 is correct. And again, if you apply the same logic, the logic which you applied in the second statement, if you follow the same logic, you'll come to a conclusion that 3 is correct. So why this question should be attempted? Because there is no option 1, 2 and 3. अगर यही क्वेश्चन में अगर 1, 2 and 3 ऑप्शन रहता तो मैं इसको अटेम्प्ट नहीं करता, बिकॉज मेरे पास हमेशा एक मन में डाउट रहेगा कि क्या पता इसको यूज़ करके हम एज भी डिटरमिन कर सकते हैं. बट सिंस दैट ऑप्शन इज नॉट देयर, right, and I know that one of the statement is wrong, or at least one is wrong, at least one is correct, then I can use this and I can find the correct answer. So in such questions, apply such logic and you will realize that your accuracy percentage keeps on improving. This for me is a little bit difficult question. Aerial metagenomics. सबसे पहले तो एक कोई भी पिछले 2-3 साल का न्यूज़पेपर उठा के देख लो, 2023 से पहले आपको ये टर्म नहीं मिलेगा. मेटाजेनोमिक्स अगर आपको पता है कि क्या है, देन यू कैन आंसर दिस क्वेश्चन. मेटाजेनोमिक्स इज दैट यू कलेक्ट अ सैंपल. आप फॉरेस्ट गए, आपने एक मिट्टी उठाया, वहां से आपको कुछ दिख रहा है कि बहुत सारा यहाँ पे कुछ रॉटिंग चीज़ है, आपने वो पूरा सैंपल कलेक्ट किया, आप लैब में लेके आए. ज़रूरी नहीं है कि वो एक ही स्पीसीज हो, वहाँ पे हो सकता है मल्टीपल स्पीसीज हो. तो अब लैब में लेके आओ आप, और लैब में ये देखो दैट कौन कौन से स्पीसीज का डीएनए यहाँ पे है. सो दिस स्टडी, रादर देन फोकसिंग ऑन वन स्पेसिफिक स्पीसीज, you focus on the entire sample. You collect a larger sample and then you analyze that larger sample, that what all species are possible here. You can do the same thing in water also, that you go to some lake, some river, you pick some water from multiple places. So without even going that कौन सा fish, कौन सा reptile यहाँ पे है, कौन सा amphibian है यहाँ पे, किस टाइप के algae हैं, without going that आपने खाली पानी कलेक्ट किया है, उस पानी से आप आइसोलेट कर रहे हो डीएनए सैंपल को और आप देख रहे हो कि कौन कौन से स्पीसीज हैं. So such type of study where the focus is not on individual species first. The focus is on first collecting a sample, then analyzing the sample simultaneously to know about all the possible DNAs which are present belonging to different species. These studies are called metagenomic studies. So metagenomics is actually a well established branch. It is used in crime also, in detecting crime. जैसे मान लो आपने देखा होगा, अगर आपने CID देखा है, तो CID में क्या करते हैं कि दया ये दोनों DNA सेम लग रहे हैं, देखा आपने वो. तो so anyways, so when क्राइम सीन पे जो क्या होगा, वहाँ पे सैंपल कलेक्ट करेंगे, कि जो भी पूरा क्राइम सीन होगा वहाँ से बहुत सारे सैंपल कलेक्ट करेंगे. एंड इफ़ यू हैव लेट्स से अ DNA डेटा बैंक, then you can match all the DNA which you are finding here with your DNA database to know that if we can find about the culprit, right? India is working on this capability. We are working on this capability. America and many European countries have been using this capability for quite some time now. Some researchers have proposed that we can collect this sample from air also, particularly in forest areas जहाँ पे wildlife बहुत ज़्यादा है, that I can collect sample from air also, and particularly to know about pathogens and microscopic organism, that aerial sample can be of great help, right? So this study is called aerial metagenomics. Hence the answer is A here. Collecting DNA samples from air in a habitat at one go and then analyzing that sample and trying to figure out all the possible DNA here. It can also tell you about pollens. आपने कुछ pollens भी collect किया है, तो आपको प्लांट डीएनए के बारे में भी पता चल जाएगा, आपको पैथोजेंस के बारे में पता चलेगा. सो प्रेजेंस ऑफ प्लांट्स, पैथोजेंस, मे बी सम बर्ड feathers have been collected, presence of birds also. You can determine many things by gathering genetic sample from air. Hence the word aerial metagenomic. But I will repeat, this is not an easy question. ये अगर आपको स्पेसिफिकली जैसे मेटाजेनोमिक एटलीस्ट अगर पता था तब आप आंसर कर सकते हो, बट अगर मेटाजेनोमिक्स भी नहीं पता है कि क्या है तो फिर इसको अटेम्प्ट करने का कोई ज़्यादा मतलब नहीं है. डी को आप एलिमिनेट कर सकते थे, सेंडिंग ड्रोन्स टू इनएक्सेसिबल एरियाज टू कलेक्ट प्लांट एंड एनिमल सैंपल्स, ये एलिमिनेट कर सकते थे, बट बाकी एलिमिनेट करना जो है मुश्किल है. And I did not, why I did not like this question, because this topic in last 1 or 2 years was covered in this very famous magazine called Economist. Economist is like one of the most read magazine in the world, but then it is only read, it is not read for UPSC preparation. Simple कि आपको UPSC preparation के लिए आपको बिल्कुल कभी भी पढ़ने की जरूरत नहीं है. It's a very famous magazine. Somebody clearly was reading this magazine, they came across this article and made a question based on it. तो ये अच्छी आदत नहीं है कि आप अपने विम्स एंड फैंसीज पे कुछ भी आप कहीं से भी पढ़ रहे हो. और ये इन्वायरमेंट में और साइंस टेक में भी हो रहा है, इन्वायरमेंट में तो और ज़्यादा हो रहा है. जैसे कार्बन मार्केट वाला क्वेश्चन सेम ईयर में आया था, इन्वायरमेंट का डिस्कशन हुआ है अभी. तो अब इन्वायरमेंट का जब डिस्कशन होगा, 2023 में कार्बन मार्केट पे क्वेश्चन था. It was also covered in this same magazine. So you can see clearly that somebody was reading this and made a question based on that. But again, don't worry, just because some questions were asked from this magazine, कहीं से मैं ये नहीं बोलने की कोशिश कर रहा हूँ कि आप इकोनॉमिस्ट खरीद के पढ़ना शुरू कर दो. Please don't do that. Not at all required. ऐसे अगर 1-2 क्वेश्चन नहीं भी अटेम्प्ट करोगे क्या फ़र्क पड़ता है. That's always a good strategy to skip few questions. And the same year there was one more question, microsatellite DNA is used in case of which one of the following. So अब ये भी एक बहुत ही specific word है, but I do feel like that looking at the option, जैसे aerial metagenomic में perhaps D आप eliminate कर सकते थे, but यहाँ पे ज़्यादा options eliminate हो सकते हैं. अब microsatellite meaning word का मान लो आपके पास meaning clear नहीं है आपको कि microsatellite का क्या मतलब है, but फिर भी let's look at the option. Studying the evolutionary relationship among various species of fauna. तो ये तो हमको पता है कि DNA का एक जो बहुत इंपॉर्टेंट रोल है DNA स्टडी का वो ये है. I know that this can be done with help of DNA. Stimulating stem cells to transform into diverse functional tissues. We are essentially talking about producing induced pluripotency stem cell, iPSC. उसमें DNA as such doesn't make a lot of sense. Promoting clonal propagation of horticulture plants doesn't make sense here also. Assessing the efficacy of drugs by conducting series of drug trials in a population, that is the most made up statement. So having the benefit of hindsight outside examination hall, I'm making this statement that perhaps B and C and D can be eliminated. The correct answer is A. Right. So microsatellite DNA is simply a very small fragment of DNA that we have many base pairs, but the smallest fragment with not more than, let's say, 9 base pairs, I call it microsatellite DNA. And even that microsatellite DNA is more than enough to tell you uniqueness about your genetic structure. At least, for example, for parental test it is used. कि ये बहुत बार होता है ना, कोर्ट केसेस में जाता है that कोई किसी ने कर दिया कि ये मेरे फादर हैं, and then microsatellite DNA can be used for that, right? And it can also allow us to understand the relationship between different organisms within a population, but also different species we can find this relationship. So answer here is A. So we use this word microsatellite, mini satellite and macrosatellite. As we cover more and more base pairs, we can gain more and more information. But at least for that first part, that studying the evolutionary relationship among various species of fauna, even microsatellite DNA is enough. So correct answer here is A. Let's quickly go through this topic of health also. H1N1 virus is sometimes mentioned in the news. देखो ये पुराना news है. H1N1 जो swine flu epidemic था, that happened, I remember when I was in college, and we all were wearing masks at that point of time also. So it was very popular, like swine flu. This is essentially a type of influenza virus which was spreading much, much more rapidly. So only if you knew that this H1N1 is swine flu, not bird flu, not clearly it's not AIDS, not dengue. But I do remember many people like getting this wrong, simply like marking on bird flu. H5N1 is called bird flu. H5N2 is called bird flu. Swine comes from pigs, that it came from their जो पूरा transit. It is a zoonotic disease, and जो primary host जो था, जो human beings से जो infection था, that occurred because of swine. Hence the word swine flu. जो H5N1 influenza virus है, that comes from birds. Hence the word bird flu. So this is a factual question. इसमें कोई ज़्यादा logic है नहीं. But this was a very popular fact. Like ideally, a candidate should know that H1N1 is swine flu, and at least in 2015 they clearly should have known, because it was hardly like, although it 4-5 साल पुराना न्यूज़ था ये, but फिर भी ये continuously न्यूज़ में था. ऐसा नहीं है कि खत्म हो गया था. It was continuously covered because influenza virus जो है इतना rapidly evolving virus है कि अब भले आप इस साल इन्फेक्ट हुए, आप अगले साल भी इन्फेक्ट हो गए उससे, कि अब नया स्ट्रेन है. वो जो नया स्ट्रेन है वो आपको फिर से वहीं सर्दी खांसी करेगा, भले आप पिछले साल आप दूसरे स्ट्रेन से आप इन्फेक्ट हुए थे. Zika virus was in news a lot. In tropical regions, Zika virus disease is transmitted by the same mosquito that transmits dengue. That is true. In fact, even chikungunya is transmitted with the same mosquito. That mosquito is called Aedes aegypti. So this Aedes aegypti is responsible for transmitting Zika, chikungunya, and dengue. Right. So first is true. But Zika का एक और जो प्रॉब्लम था, सेक्सुअल ट्रांसमिशन ऑफ जीका वायरस डिजीज इज पॉसिबल, दैट इज ऑल्सो ट्रू. इनफैक्ट जीका का एक और बहुत बड़ा प्रॉब्लम ये है दैट इन्फेक्टेड मदर कैन ट्रांसमिट दिस टू अ बेबी, एंड द बेबी वॉज बॉर्न विद दिस like some developmental function will be restricted because of small brain size, and I think there is a word for it, microcephaly, something like that. I am forgetting the word, but this, sorry, microcephaly is called. So microcephaly जो है, that was a disease which occurred from pregnant mother to child because of Zika virus. So this was in news a lot. Right now it is not in news a lot, but this was, जैसे right now अभी कौन सा अभी flu news में था अभी? Which flu was news? केरला में जो अभी बहुत सारे केस, केरला में तो नहीं, but अफ्रीका में जो केसेस बहुत सारे आए थे अभी, मंकी फ्लू, मंकी पॉक्स. जो मंकी पॉक्स है, जैसे अगर मुझे अगले साल के करंट अफेयर्स के लिए कुछ पढ़ना है डिटेल में, सो आई विल रीड अबाउट मंकी पॉक्स. तो ये जो क्वेश्चंस हैं, दे ऑल्सो गिव यू अ हिंट दैट व्हाट टाइप ऑफ न्यूज़ यू आर सपोज टू स्टडी. सो आई डू नॉट हैव टू वरी अबाउट जीका वायरस. UPSC, remember, can always ask question on any theme what they want, but ideally they will pick themes from current affairs. So that monkeypox is much more important. Multi-drug resistance as a topic was in news a lot for many years. It will remain in news. UPSC has asked questions in mains examination at least twice in last 10 years, and they have also asked questions in prelims examination, I would say directly or indirectly twice or thrice. जैसे अभी बायोफिल्म वाला क्वेश्चन था, वहाँ पे भी मल्टी ड्रग रेजिस्टेंस के बारे में बात थी. Which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India? I remember many institutes getting this wrong. And I was not able to come to a conclusion that why people would think that 2, 3 and 4 is correct. Like why would you think that 4th is correct here? तो जो multi-drug resistance है, पहले तो ये है कि microbial organisms such as bacteria or fungi or worms or even viruses, they have developed some sort of immunity against the very medicine we take to neutralize them. And ये problem सारे pathogens में है, but bacteria में सबसे ज़्यादा बड़ा problem है. और उसका जो सबसे बड़ा cause है हमारा erratic use of antibiotic है. देखो ये सबसे पहले तो बहुत clear होना चाहिए in everybody's mind that all species evolve, and species who multiply more rapidly, and even though it is asexual reproduction, the chance of them developing a strain is higher. So when there is a evolutionary pressure on your survival and reproduction, then mutations which is going to give you some extra advantage will be selected by evolution much more rapidly. So bacteria are also evolving. We have found ways to kill bacteria which can cause us diseases with help of antibiotic. But if we are taking lot of antibiotic when it is not required, then now how things happen? Many bacteria are simply neutral for us. Bacteria बहुत बदनाम है, but remember बिना बैक्टीरिया के लाइफ पॉसिबल नहीं है, राइट? बिना बैक्टीरिया बिना फंगाई के लाइफ पॉसिबल नहीं है. But most of the bacteria are neutral, some are harmful, and many of them are useful, right? जैसे we cannot digest food without our gut bacteria. There was a question on that also in UPSC. So if you consume excessive antibiotic, then you kill. Imagine that some bacteria which have some strain which has more resistance against antibiotics. When you take erratic antibiotic whenever it is not required, then you kill many. जिस तरीके से species compete करते हैं resources के लिए, bacteria भी compete करते हैं, and that competition controls their population. But if you take excessive antibiotic, then you are eliminating the competition of those bacteria which already have some sort of immunity against it, and now they will multiply much more rapidly. So the prevalence of this drug resistant bacteria increases. So we can logically understand that how does this function. But it can also occur if you are not utilizing your antibiotic effectively, which may seem paradoxical. I am saying that excessive and underutilization both is a problem. आपको डॉक्टर ने 5 दिन का एंटीबायोटिक दिया था, अब 2 दिन में ठीक हो गया, आपने छोड़ दिया. Maybe the bacteria has weakened right now. It has not died. But in this now, this is a evolutionary pressure of the survival. In this evolutionary pressure, that mutation which can increase their chance of survival, that probability increases. So if a bacteria is weakened but not dead, and you have stopped antibiotic, then again there is also a chance that a strain can develop. So if you are not suffering from a bacterial infection, then ideally you should not take antibiotic. बहुत बार डॉक्टर्स, जब तक डॉक्टर ना बोले तब तक तो लेना ही नहीं है. और डॉक्टर देखो वायरल इन्फेक्शन में एंटीबायोटिक आपको तभी देंगे जब आपका वायरल इन्फेक्शन बड़े दिन से ठीक नहीं हो रहा है. वायरल इन्फेक्शन का कुछ कर नहीं सकते आप, बॉडी फाइट करेगा वो. बस ये है कि आप बढ़िया खाना खा सकते हो, पानी पी सकते हो, ये सारी चीज़ें कर सकते हो. उसमें क्या होगा कि आपका बॉडी का इम्यून सिस्टम कॉम्प्रोमाइज्ड है, तो जो बैक्टीरिया जो जनरली आप आराम से लड़ सकते हो, आप नहीं लड़ पाओगे. तो ऐसे केसेस में बचने के लिए डॉक्टर एज अ प्रिकॉशन आपको एंटीबायोटिक देंगे. बट जनरली जैसे ही थोड़ा सा बुखार हुआ, आपने एंटीबायोटिक ले लिया, डॉक्टर से भी प्रिस्क्रिप्शन नहीं लिया, आप गए आप काउंटर पे, उन्होंने आपको एंटीबायोटिक दे दिया. दिस थिंग्स आर द बिगेस्ट कॉज ऑफ ड्रग रेजिस्टेंट बैक्टीरिया. बट डूइंग द सेम थिंग इन पोल्ट्री एंड बवाइन एंड स्वाइन इज अगेन गोइंग टू कॉज एंटी रेजिस्टेंट बैक्टीरिया. अब जैसे केरला गवर्नमेंट ने अभी रिसेंटली एक बहुत बढ़िया गवर्नमेंट जो है इंस्ट्रक्शन आया दैट जितने भी एंटीबायोटिक्स हैं, you cannot buy them and nobody can sell them without a proper doctor's prescription. अब इसका भी एक चैलेंज ये है that ये जो ड्रग रेजिस्टेंट बैक्टीरिया प्रॉब्लम है, ये ऐसा नहीं है कि कोई स्टेट स्पेसिफिक प्रॉब्लम है. सो अनलेस many countries together who share a boundary invest into that, this problem will not go away. So forget about states, forget about all states working together. So it shouldn't happen just in Kerala, it should happen throughout the country. It should happen throughout the world that erratic uses of antibiotics should be restricted. So in any case, the correct answer here is 2 and 3. Genetic predisposition of some people, no. Multiple chronic diseases in some people, no. The answer is 2 and 3. Other reasons can be if there is a place where bacteria can multiply rapidly. What are those places? Where there is a lot of organic waste. Organic waste सबसे ज़्यादा कहाँ पे होगा? Waste site पे होगा, hospitals में होगा. So hospitals, waste sites, all of these are again places where this evolution of bacteria can happen more rapidly and they can develop a strain which has resistance against antibiotics. So if you go to WHO, World Health Organization, they clearly mention all the causes. Overprescribing of antibiotic, patients not finishing their treatment, that is also a cause. Overuse of antibiotic in livestock, poor infection control in hospitals and clinics, lack of hygiene and poor sanitation, lack of new antibiotics being developed, which is also a challenge, right? So these are the reasons why this is a issue. यहाँ पे जो question का answer था वो 2 and 3 only है. B is the correct answer. This was a question again inspired from current affairs. Hepatitis was continuously in news. Hepatitis is a liver infection caused by many type of viruses. A, B, C, D, E, at least we know 5 different strains. यहाँ पे पूछा है not correct, तो 3 करेक्ट है, 1 इनकरेक्ट है इसमें. ये जो बी है बहुत फेमस है, हेपेटाइटिस सी का जो है कोई वैक्सीन नहीं है, ये बहुत फेमस स्टेटमेंट है. बट बी इज सेइंग दैट हेपेटाइटिस बी डज नॉट हैव अ वैक्सीन. स्टेटमेंट बी क्या है? हेपेटाइटिस बी अनलाइक हेपेटाइटिस सी does not have a vaccine. The exact opposite is true. Hepatitis C does not have a vaccine. Hepatitis B does have a vaccine. So only if you knew this, then you can get this answer correct. But चलो फिर भी देखते हैं कि क्या पता हम बाकी, अगर हमको नहीं भी पता था ये चीज़, can we eliminate other options? Hepatitis B virus is transmitted much like HIV. So essentially we're talking about sexual transmission or transmission due to infected blood or semen, etc. So that is actually true. Hepatitis B can be transmitted sexually. C is also true. That globally the number of people infected with hepatitis B and C viruses are several times more than those infected with HIV. उसका बहुत बड़ा कारण ये है that hepatitis जो है it can remain inactive for many years before it starts showing symptom. तो आप infected हो but आपको पता भी नहीं है. Some of those infected with B and C viruses do not, हाँ ये दे भी रखा है, show the symptom for many years, तो ये भी दे रखा है. So जो correct, although so I don't think that it's easier to eliminate if you do not know that जो option B है वो गलत है. So option B यहाँ पे गलत है and hence in this question that is the correct answer. Pneumococcal conjugate vaccines. The conjugate vaccines are also this very specific जो sub unit approach मैंने discuss किया था. And pneumococcal bacterias are group of bacteria known to cause some diseases. जो सबसे famous है वो pneumonia ही है. But even diseases like meningitis and other diseases can occur due to pneumococcal bacteria. So these vaccines are effective against pneumonia as well as meningitis and sepsis. These are diseases caused due to this bacteria, so that makes sense. Dependence on antibiotics that are not effective against drug-resistant bacteria can be reduced. That also makes sense. That if antibiotics are not active against these bacteria, but you have taken these vaccines, then your body has developed this immune response against these bacteria. तो हमारा जो antibiotic पे dependence है वो हमारा कम हो गया. But third is not true for any of the vaccine. ये दुनिया का कोई भी वैक्सीन हो, ये कह देना कि कोई रिएक्शन नहीं होगा, that these vaccines have no side effects and cause no allergic reaction. ये दुनिया में कोई भी वैक्सीन ऐसा नहीं है जिसके लिए स्टेटमेंट ट्रू है. All vaccines can cause, again वो रिएक्शन अलग अलग हो सकता है, किसी के लिए हो सकता है कुछ भी ना हो. But saying that everybody is completely immune to any allergic reaction by any of the vaccine, I'm saying that it is, forget about this. For any vaccine, that statement is wrong. So third statement is wrong. First is correct, second is correct. Answer is 1 and 2 only. Conjugate vaccine is a specific vaccine where there are certain bacteria, they can outwit immune system of a young children. That उनका जो bacterial pathogen part है, जो protein है, जो antigen है, it is coated in a layer of sugar. And that layer of sugar जो है वो body का immune system उसको फिर वो पकड़ नहीं पाता है. And because of that, it does not think of this as a pathogen and does not think of this, does not produce antibodies against it, right? And in pneumococcal conjugate vaccine, then we take care of that. So that conjugate vaccine is for specifically those bacteria which can outwit immune system of young children. So we train body from very young ages how to identify such type of bacteria. This is again a question due to COVID-19, current affairs. ACE2, ACE2. ACE2 is part of naturally occurring enzyme in many organisms, including human beings. ACE2 play a very important role in many things. Why they were very much in news due to COVID-19 pandemic? That the structure of this virus, it perfectly matched to the receptor of ACE2. And it immediately, the moment you are infected, the virus is inside your body, it immediately made a complex with this enzyme ACE2, and it immediately entered inside your cell, and your body has no chance of fighting this pathogen from outside. So literally, there is an enzyme inside this body which is calling it in once you are infected, right? So ACE2, and by the way, this actually was one of the reasons why many people still think that this was simply an engineered virus. That ये जो structure similarity hai, that this spike protein and that ACE2 enzyme structure, it matched so perfectly that naturally happening, that ये naturally हो गया हो evolution के कारण, इसका probability बहुत कम है, zero है. And that is why many people believe that it is a engineered virus, lab engineered virus. Even today, many people believe that this is a lab. There are many other evidences, including the fact that there is a lab in Wuhan called, there is a lab of virology in Wuhan where this pandemic emerged. So anyways, it was in news because of spread of viral diseases, particularly COVID-19 viral disease. So that is why this question was asked in 2021. Another question, yes, ये टॉपिक कोविड-19 के कारण वायरस, वायरल इन्फेक्शन, वैक्सीन, ये सारा जो थीम है बहुत बार लगातार आया था. So adenoviruses have single-stranded DNA genome, whereas retroviruses have double-stranded DNA genome. Common cold is sometimes caused by an adenovirus, where AIDS is caused by retrovirus. ये question कहीं से आसान नहीं है. Only when you know about details of virus structure, their genome, and not just genome, also their replication, how they infect a host and then replicate. Only when you know that, only then you can answer this question. जैसे जो retrovirus हैं, these are RNA viruses. तो पहला statement तो वहीं से गलत हो गया. The retrovirus have double-stranded DNA genome. ये statement गलत है. By the way, वो भी गलत है. Single-stranded DNA genome, no. Adenovirus is double-stranded DNA genome. So that statement is also wrong. And retroviruses have double-stranded DNA genome, no. Single-stranded RNA genome. Why we call them retrovirus? Because when they infect a host, then they convert this into a complex DNA molecule. And with help of that DNA molecule, now they infect the host and they multiply and they go on to infect other cells. Unlike other RNA, so this is also RNA virus, but it is not same as coronavirus. Coronavirus was RNA virus, but coronavirus को करने की जरूरत नहीं है. So they are called retro because remember what I discussed in the starting of the class, that in living organisms you have DNA transcribes into RNA. That is a sequence which occurs in most of the organism. यहाँ पे क्या हो रहा है? Opposite हो रहा है. It is happening in the reverse direction. There is a RNA virus which is leading to a corresponding DNA molecule. So this is happening in reverse direction, hence the word retrovirus. And coronavirus is not an example of retrovirus, even though it is an RNA virus. But AIDS का जो virus है, that is an example of retrovirus. And yes, adenovirus is not that dangerous, but adenovirus can cause things like common cold. It is much more common around us. So 2 only is the correct answer here. First statement is wrong and first statement का दोनों part wrong है. पहला भी wrong है और दूसरा भी wrong है. But I will repeat, ये कहीं से भी आसान question नहीं है. Only when you know the details of virus and their structure and also how they replicate, only then you can answer this question. There is a question on probiotics. Probiotics are made of both bacteria and yeast. आप जाओगे shops में, you'll find this probiotic food. तो probiotic food क्या है? एक दो टाइप का फूड होता है, एक होता है प्रीबायोटिक, एक होता है प्रोबायोटिक. सो प्रोबायोटिक में यू आर इसेंशियली टेकिंग गुड बैक्टीरियाज. बट कुछ फूड ऐसा होगा जिसको हम जब प्रीबायोटिक बोलते हैं, इट हेल्प्स इन प्रोडक्शन ऑफ गुड बैक्टीरियाज इनसाइड योर बॉडी. जैसे आप जब फाइबर वाले फूड खाते हो, जैसे एप्पल को आप छिलके के साथ खाते हो, सो दैट हेल्प्स इन योर डाइजेशन. ऑर्गेनिजम्स इन प्रोबायोटिक्स आर फाउंड इन फूड वी इंजेस्ट बट दे डू नॉट नेचुरली अकर इन आवर गट. ये बिल्कुल ही गलत है. हमारे गट में बिलियंस ऑफ बैक्टीरियाज हैं एंड विदाउट दैट गट बैक्टीरिया नो फूड कैन बी डाइजेस्टेड. सो our gut in itself is a very complex ecosystem where along with our internal organs you also have many bacterias, and these bacterias help us in digestion of food. In fact, bacteria also help in production of some type of biomolecules. Specific vitamins, for example, are produced with help of these bacteria, and without them again we will suffer from various type of diseases. So these bacteria are helpful for us. And there is a remarkable way how our body does not recognize them as a pathogen. That है बैक्टीरिया, बट हमारा बॉडी इनको दोस्त समझ रहा है कि ये दोस्त हैं भी. और या सबसे अमेजिंग, नेचर इतना अमेजिंग है, ये होता है मदर्स मिल्क के कारण. दैट व्हेन अ बेबी ड्रिंक्स मदर्स मिल्क, that mother milk contains that bacteria and gradually trains the body that these are good bacterias, and when as you grow up your body learns to live with these bacteria, and these bacteria they help us in digestion. So second statement is not at all correct. Third is correct. Probiotics help in digestion of milk sugars. So that is correct. Answer here is 1 and 3. So your gut is hosting many type of bacteria, and these bacteria are helping us in digestion of various type of food. There is a question on B cells and T cells. This is again due to COVID-19 pandemic and virus and immunity. So understanding the immune system. हमारा immune system बहुत complex है और बहुत amazing है. आपको कोई भी पैथोजन पहले अटैक करे, कुछ कुछ रिस्पॉन्स जो होगा बहुत ही सेम होगा. दैट एनी टाइप ऑफ पैथोजन सिमिलर रिस्पॉन्स विल अकर. आपको बुखार हो जाएगा. बॉडी इज ट्राइंग टू रेज द टेम्परेचर. सर्टेन molecules are there because many bacteria, many pathogens cannot survive beyond 40 degrees Celsius. Some can. So अगर ये काम नहीं करे, बुखार हो गया, कुछ और चीज़ें किसी और, so similar से response होंगे. But if that does not work well, then your this what we call adaptive immunity comes into. So this is called innate immunity. But then this adaptive immunity comes into place, where in the bone marrow you have these specific cells, B cells and T cells, which helps us in making specific antibodies to kill specific antigen. तो B cell का response immediate होगा, T cell का response थोड़ा देर बाद होगा, but वो ज़्यादा लंबे टाइम तक टिकेगा. So the purpose of B cell and T cell जो हैं, they are part of our adaptive immune system, which helps us in producing specific antibodies to kill antigens. So answer here is D. They protect the body from the diseases caused by pathogen. D is the correct answer. And again, this is also a very expected question. This was covered a lot in newspapers, and this was covered a lot during COVID-19 pandemic, that how body's immune system work. And one last question in this topic of health. This is one of those questions, one of those examples where a topic was in news 2.5 years ago, but UPSC asked a question later. I remember teaching this topic in 2018, 2019, but after that I haven't taught this topic, Wolbachia method. But this question was asked in 2023. So I was able to find at least one reference of this method in 2021, November 2021, but this question was asked in 2023. Right, so this is a topic, again, if you only know it, then in 2020, 2019, this was a very easy question, but in 2023, this is a difficult question simply because नाम से देखो कुछ भी पता नहीं चलेगा. तो होता क्या है? Let me briefly explain this. Wolbachia is a group of bacterias, and these bacteria naturally occur in many living organisms, including human beings. They do not cause any harm to us. So we are not affected by Wolbachia bacteria. But they are harmful for mosquitoes, particularly that Aedes mosquito which I talked earlier, which spreads Zika, dengue, and chikungunya. So when Aedes mosquito is infected with Wolbachia, then its viral load reduces. It's viral, that virus which causes these diseases, that virus holding capacity decreases. So if you infect both male and female mosquito with Wolbachia, then all the babies which are produced will have again the bacteria in their body, and then this capacity to carry this virus as a pathogen that reduces, which reduces the burden of all of these viral diseases which we talked about. So in some countries this has been already tested, that does it function, and actually does function really well. And in countries such as some African nations, this was tested. Even in Bangladesh, if I remember, there was some So a lot of mosquitoes were captured, infected into a lab, and then they were released into atmosphere again. And then the goal here was to understand that if the infectivity of these viral diseases, which occurs because of Aedes, is it decreasing? And it did show positive result. So answer here is controlling the spread of viral diseases by mosquitoes. But yeah, if you did not know this in 2023, then my recommendation will be that do not attempt this question. If you, in 2025, when you come across a question like this, and maybe it was in news in 2021, simply you haven't read that, so simply skip that question. That's always acceptable. Okay. Any questions? If you have till now, you can ask questions. Let's take a break here then. After that, we will discuss topics of ICT and the remaining topics of science and technology.
Does the vast syllabus of general studies for the UPSC prelims overwhelm you to initiate your preparation for the civil services examination? Is your apprehension and self-doubt coming in the way of your success in joining the civil services? Comprehend your preparation for UPSC Prelims 2025 with the fast-track course targeting general studies with innovative curriculum, revision and mentorship. Starting from 19th November at 1 p.m., the course offers coverage of UPSC GS Prelims subjects, dedicated mentorship support and guidance for performance maximization, access to PT 365 classes for current affairs preparation, sectional mini tests for regular assessment and performance, personalized student portal to access study material, access to live and online classes. Enroll today and make prelims your stepping stone to proceed to the UPSC mains.
The next set of topics are from ICT. Many questions from current affairs not at all relevant right now. For example, in 2015, there was a question, Project Loon, sometimes seen in the news, is related to. तब ये न्यूज़ में था, this project has been abandoned by Google. ये Google का project था. The goal here is that with help of these floating balloons in upper layers of troposphere or lower layers of stratosphere, we will provide internet in remote and rural areas. But in 2020, they said that this is economically not feasible, and then they abandoned the project. So knowing this, that it was this wireless internet transmission in rural areas, if you know this, then you know that this is B is the correct answer. A, C, D are not correct. ये तब important था, बिल्कुल important अभी नहीं है, but it tells you that what type of questions can be asked. Near field communication. Advancements in communication has always been a favorite topic of UPSC. It is a contactless communication technology that uses electromagnetic radio fields. So when you go and use your debit or credit card on a POS machine, then these new cards do not have to be inserted inside the machine. You just tap your card and the money gets automatically deducted, isn't it? And to do that, it essentially utilizes radio waves to share information between this card and this POS machine. So first is true, but the information which is transferred, that occurs with help of radio waves. The second is that NFC is designed for use by devices which can be a distance of even a meter from each other, not at all. ये जो है बहुत ही important fact है, that NFC works within few centimeter, ideally 3, 4, 5 centimeter, at max 10 centimeter. There was a reason why it was done like this. With help of NFC, we also wanted to ensure that even if you want to send, let's say, some encoded personal data, then it can be transferred. We do not want this to be a long range device. And even so, long range, short range का कोई meaning नहीं है जब तक हम एक compare ना करें. But we want, जैसे for example, जो सबसे famous application NFC का है, वो card payment में है. And then we do not want this to happen at distance of 1 meter or 5 meter. So this is totally wrong. In fact, even a meter, it specifically has been designed to work less than, at range of about less than 10 centimeter. And this is, by the way, if you know NFC, then you know that the second statement is a very popular थिंग, मतलब ऐसा हो ही नहीं सकता कि आपने NFC के बारे में पढ़ा है और आपने ये नहीं पढ़ा हो कि इसका जो रेंज है वो 10 सेंटीमीटर मैक्सिमम रेंज है. Third is true, can use encryption when sending sensitive information. Depends upon what type of information it is. For example, if it is your credit card data, then it needs to be transmitted in encrypted form. That is, को decrypt करना आसान ना हो. So third is true, that we can use encryption when sending sensitive information. Answer is 1 and 3. Answer is C here. That range part was wrong. And even though you may think that it is a factual statement, but it is a very prominent fact about near field communication technology. Another question on communication technology, this time on Li-Fi, light fidelity. In Wi-Fi we use wireless fidelity जो है, जो Wi-Fi router यहाँ पे होगा कहीं पे इस room में. Wi-Fi router uses radio waves particularly of 2.4 gigahertz, 5 gigahertz, 6 gigahertz. These radio waves are used for transmission of data. We call it Wi-Fi. Li-Fi, which was demonstrated for the first time, I think in 2013 by a person called Harold Haas in a very famous TED talk. He delivered a TED talk about 10 years ago. And in that TED talk, he demonstrated that with help of visible light, it is possible to transmit data across a small distance. He did that for small distance. We want to make it for large distance, although there are some challenges. I'll come to that part. So how does it work? We use LED bulbs, and these LED bulbs, they essentially pulsate very rapidly. That these LED bulbs, jo intensity hai, it fluctuates very rapidly. In 1 second, it can fluctuate million times or billion times. And that fluctuation is so rapid and so small that our eyes cannot make sense of it. So there can be a Li-Fi device in this room, and by looking at it, you will not know. It is like any other LED bulb for our eyes. But that pulsating is not random. That intensity fluctuation is not random. It is following a pattern. And then there is a receiver here. And that receiver can intercept how much of photons are changing. And essentially with help of that pulsating intensity, we aim to transmit those zeros and ones, those binary signals. And that receiver, which often will be a photo detector, a light detector, will detect that and will convert that into a corresponding electrical signal and will receive this data. There are really good videos available. In fact, आप simply type करना जाके Harold Haas Li-Fi, the very first TED talk from 11 years ago. You'll find this video, or I think 30, I don't know exact year, but it is definitely more than 10 years ago. You'll find him demonstrating transfer of a picture with help of LED bulb. And you'll find another TED talk from more recent so he was demonstrating how solar panels can be used for this particular purpose. And Li-Fi is many times faster than Wi-Fi. Although Wi-Fi 7 जो है, जो सबसे latest Wi-Fi होगा, जो अभी हम use नहीं कर रहे हैं, but Wi-Fi 7 का speed बहुत ज़्यादा है. Like the speed is supposed to be 20 GB, 30 GB per second, something like that. Li-Fi is comparable to that. But in 2015, when this question was asked, we used Wi-Fi 2nd or 3rd generation. And without a doubt, it is 10 times, 20 times more faster than any Wi-Fi. So यहाँ पे दोनों statements सही हैं. That it uses light as medium for high speed data transmission. It is a wireless technology and it's several times faster than Wi-Fi. Answer is both 1 and 2. A very similar question was asked in 2020 also. Li-Fi is also called visible light communication simply because we are using visible light to send data. 4th statement question. If you look at the options, you see that one of the statement is wrong. Statement dekh lo, only one is wrong. VLC uses electromagnetic spectrum wavelength 370 to 780 nanometer. So 375 to 700 है वो visible light है. 780 तक what we call shortwave infrared, but that is also fine. Li-Fi does use infrared also. First statement में कोई problem नहीं है. Second statement is not at all correct. That it is a long range optical communication. Think about this setup. It is based on a pulsating LED bulb. So let's say that I have this LED bulb here which is acting as a Li-Fi device. And I have some photodetector at the end of this room which is able to intercept this change in pulsation. It is capable of doing that, no problem there. But the moment if this photodetector is outside this wall here, which is closer to that place, that signal will be disrupted because this wall is opaque. Visible light cannot cross through opaque things. Radio wave can. Most of the low frequency radio waves can easily cross through solid objects. Visible light cannot except glass. Glass is transparent. Most of the solid objects are opaque for visible light. That is a problem. So hence it cannot be called long range optical communication. If I know this, then I know my answer is 1, 3 and 4. But look at the other options also. VLC can transmit large amounts of data faster than Bluetooth. That is true. VLC has no electromagnetic interference. This is not a very well framed statement. Although I will consider this to be true, but the complete statement will be that VLC has no electromagnetic interference from radio devices. So कहीं पे भी आप चलो, आप Li-Fi के बारे में पढ़ो. पहले तो why most of the devices which we use in our day-to-day life, think of Wi-Fi, think of mobile phone, they are operating in the range of radio frequency. Li-Fi is visible light communication is operating in the range of visible light frequency. कोई match नहीं है frequency का. Visible light frequency is 100,000 times more than frequency of radio wave. So if you are using a Li-Fi, neither it will affect your radio devices, nor it will be affected by radio devices. That is the complete statement. But can it be affected by, let's say, some other bright light? कि सनलाइट बहुत ब्राइट है यहाँ पे, या ये एलईडी बल्ब है, बट बहुत सारे बल्ब यहाँ पे और हैं जो बहुत ब्राइट हैं. हर कम्युनिकेशन सिस्टम का एक होता है ना अनवांटेड सिग्नल, नॉइज जिसको हम बोलते हैं. तो यहाँ पे नॉइज क्या है? यहाँ पे जो नॉइज है वो आउटसाइड लाइट है, दैट वेरी ब्राइट सनलाइट है. एंड इज दैट ब्राइट सनलाइट इलेक्ट्रोमैग्नेटिक वेव? ऑफ कोर्स लाइट इज एन इलेक्ट्रोमैग्नेटिक वेव. सो दैट इज वेयर दिस स्टेटमेंट इज प्रॉब्लमेटिक. जिसने भी बनाया था उसने वो चूज तो किया ये पार्ट, बट उन्होंने कंप्लीटली नहीं लिखा इसको. That VLC has no electromagnetic interference from radio devices. That will be more appropriate option. But I will still take that. I will still take this because second cannot be true. It cannot be long range optical communication. That is in fact the biggest challenge in Li-Fi, that it cannot cross walls. It cannot be a long range communication. Answer here is 1, 3 and 4. OLED display. I do not expect this question on OLED display again. जो हमारे पुराने LCD LED displays थे, पुराने जो टेलीविजन थे, LCD अभी भी मिल जाएगा आपको LCD LED display. तो जो अभी आप LED display खरीदते हो, वो एक्चुअल में the complete name should be LCD LED display. तो यहाँ पे होता क्या है? That at the back of your TV there is a backlight of white light, and on the front there are many layers, substrates, and as this light passes through, different colors are produced. लंबा लाइफलाइन होगा इसका, but जो color contrast होगा वो अच्छा नहीं होगा, and it is going to consume more power also. So it will not produce very good color contrast. In OLED, organic light emitting diode, we literally use first of all LED bulb. So if अगर आप OLED आप panel आप open करोगे, तो at the front you will find that front पे आपको दिखेगा ये red, blue, green आपको दिखेगा, और ये जो red, blue, green है, when it is illuminated in the same light, or when if it is illuminating, it receives electricity, you switch it on, it is receiving electricity. तो यहाँ पे backlight की कोई ज़रूरत नहीं है, ये LED अपना खुद का light produce कर रहा है. And if it is all three of them are illuminating in the same intensity, it will produce white light. All three of them are switch off, it will produce black light. But if red is brighter compared to, let's say, blue and green, it will produce, let's say, some other color, pink color, or some magenta color, or some orange color. So I can produce all these colors with help of this primary color. So I do not have this backlight. अब LED बनाने के लिए जो हम substrate, जो हम पूरा structure use करेंगे, if it is inorganic, then that will be inorganic LED. But a better technique is using an organic LED. Organic LED gives you a lot of flexibility. Organic LED, however, is prone to degradation faster. तो OLED का सबसे बड़ा benefit ये है that इसका color contrast best है. Nothing comes closer to OLED. इसका बस एक disadvantage ये है that ये 5-6 साल में इसका जो organic substrate है, it will start degrading and you'll have to then change your television. तो आप बहुत expensive खरीदोगे, but 4-5 साल 6 साल बाद आपको फिर change करना पड़ेगा. But if you're buying OLED, then you are quite rich, I guess. So you can do that, right? So anyways, by the way, you can also make foldable display with a fold. जो organic substrate का फायदा ये है कि organic चीज़ों को fold करना आसान है. तो foldable display बना सकते हो, stretchable display बना सकते हो. यही question था. OLED display can be fabricated on flexible plastic substrates. Roll-up displays embedded in clothing can be made using OLED. Transparent displays are possible using OLED. All three of them are correct. Answer is 1, 2 and 3. ये बड़ा ही एक फैक्चुअल सा क्वेश्चन है. मेरे हिसाब से तो अगर आप थोड़ा लॉजिक लगाओ तो शायद आप कर सकते हो अटेम्प्ट. In India, it is legally mandatory for which of the following to report on cyber security incident. तो cyber security incident के लिए जो organization responsible है इंडिया में वो है CERT-IN, CERT in Computer Emergency Response Team India. Works under Ministry of Electronics and Information Technology. As per IT Act, there is a list given that these organizations must report if there is a cyber security incident to CERT-IN. अब वो अगर आपको लिस्ट पता है तो आपको आंसर करना चाहिए. बट फिर भी अगर आप चाहो थोड़ा गेस करना चाहते हैं, ये 3 चीजें हैं: बॉडी कॉर्पोरेट, सम बिग कॉर्पोरेट, डेटा सेंटर्स, सर्विस प्रोवाइडर्स. तो अगर आपको लॉजिक लगाना हो तो आप ये लगा सकते हो. हमेशा जैसे एक और जो हम लॉजिकल रीजनिंग करते हैं प्रीलिम्स में, दैट इफ यू आर थिंकिंग ऑफ अ स्टेटमेंट टू बी फॉल्स, then why are you thinking it is false? And if it is false, then what should be correct? That reverse thinking can help you in eliminating some of the options. I will not say that it will work in every question. Just say, if there is some law, clearly there is some law, right? Option में कोई none of the above तो नहीं है. So clearly there is some law. And as per some law, some organizations are supposed to report the cyber security incident to CERT-IN, CERT-IN. If I have to guess here, then I will definitely guess 2 to be correct. Data centers are too important. Data centers handle huge amount of data that can be sensitive data. So they should definitely be in this list. So I should ideally think of that 2 should be true. But if I have to choose between 1 and 3 now, then I would choose 3. That body corporate, we are talking about large companies versus some service provider का कोई meaning भी नहीं है. Like service provider तो कुछ भी हो सकता है. Vision IS is a service provider. Right? Service provider का कोई ऐसा specific meaning नहीं है. But अगर मुझे 1 and 3 में कुछ choose करना हो, I will choose 3. That body corporate, a large company, ideally should report cyber security incident. So I should ideally think 2 and 3 to be correct, and which gives me the answer that even first is correct because there is no option 2 and 3. Answer is D. Same thing, I'm doing this post facto analysis outside examination versus doing the same thing in examination. So if you are not confident, then you can skip this question. But maybe some logic can be applied and we can think of attempting this. That is also there. Even in body corporate, it can be a small company or large company. So then you think that, does this mean that this law has some sort of distinguishing factor कि आपका revenue 500 करोड़ से ज़्यादा है तभी आप करो, 500 करोड़ से कम है तो फिर ये आप इस तरीके से जब आप सोचोगे तो आपको लगेगा कि this does not make sense, right? तो but again I'm doing this post facto analysis, but it's totally fine to skip such questions if you are not very confident about it. This is one of the lengthiest question, but one of the easiest question. When the alarm of your smartphone rings in the morning, you wake up, tap it to stop, which causes your geyser to switch on automatically. यहाँ से answer है Internet of Things. Internet of Things essentially is our day-to-day devices, which are part of a network, and the most famous network is of course Internet. And these devices all the time are gathering data about their surrounding with help of sensors. And these devices are programmed in a way that, let's say, just a simple example ले लो, that I have some thermal sensor in all of my rooms. which is all the time measuring thermal signature of human beings. And if there is no thermal signature for 10 minutes, then it sends a signal to AC to be switched off, because both of them are part of this IoT network, which is being operated with help of, let's say, my Wi-Fi router. That all of them are connected to the same Wi-Fi router, and they can send a signal to each other. So this is IoT, and I can always, of course, control these devices with an app which I have installed on my mobile phone. So this technology where devices are interconnected, and if I want not just mobile phones and laptops which are connected to internet, but our day-to-day devices like fridge or washing machine or alarm clocks or watches, these devices are connected to internet. And it's not that I am controlling these devices with some app. I can always do that. But ideally, these devices are sending signals to each other. They're equipped with many sensors, actuators, and they are communicating with each other. This technology, this setup is called IoT. So, एक line के बाद पढ़ने की ज़रूरत नहीं है. Answer here is Internet of Things. Read the entire statement. You'll get the gist that it is a description of Internet of Things. API, Application Programming Interface. It has been a revolution in how we use apps these days. एक simple सा example ले लो, that आपके पास बहुत सारे widgets हैं मोबाइल फ़ोन पे, जैसे बहुत सारे widgets हैं जहाँ पे आप weather देख सकते हो. एक simple सा example ले लो, that you are monitoring weather of Delhi, कितना rainfall possibility है, कितना temperature है. So do you think that this widget, which is being provided by some company, let's say Google, has actually invested in some meteorological satellites to know about that? Not at all. There are meteorological information available on many portals, companies which actually do that. Google invests into some protocols, and these protocols are called API, and these protocols allow a specific information to be shared between two apps. So when I use the word API, it's very simple, that there are many apps being run based on cloud-based platform. But if these apps have to send data to each other, this is where this set of protocols come into place. You can think of this as a waiter, that you order something in a restaurant, right? तो waiter जो है वो आपको order लेके जाएगा kitchen में, और kitchen में चीज़ें बन के आएगी, तो आप पूरा kitchen, पूरा menu आप order नहीं कर रहे हो, you are ordering a specific thing from the menu. Any of those things from the menu can be ordered. You are ordering a specific thing. The purpose of waiter is to bring that specific thing to you. That is what API is. That when you log in, let's say for example, somewhere with help of Google credential, बहुत सारे जगहों पे आप account थोड़े बनाते हो. आपने क्या करा है? Google account आपने use करके login करा है. तो does that mean that Google is sharing all of your data with that particular company? Clearly not. They're perhaps sharing the data about your email ID, about your phone number, your name, maybe in some cases birthday also. But it is not sharing all of your data. So the rules and protocols which allows apps to communicate with each other, we call them API. So this question is asking, Aadhaar provides open API. What does it imply? It can be integrated into any electronic device, which is true. So now you can think of this fact that UIDAI, this government agency, has this giant server where all of the Aadhaar data is stored. But how I can use this in multiple ways? मुझे DigiLocker पे access करना हो अपने ऐप पे, I can use my Aadhaar credentials. मुझे अपना KYC बैंक में कराना है, I can use my Aadhaar credentials. Does that mean when I'm doing it कि UIDAI मेरा सब डेटा इनको दे रही है, मेरा iris scan, fingerprint नहीं दे रही है? At one point of time, in maximum cases, KYC में हो सकता है कि fingerprint वगैरह करा रहे हो, but maximum cases में मेरा आधार नंबर मेरे मोबाइल नंबर से authenticate हो गया. It is being done by me. मेरा मोबाइल नंबर है, मेरा आधार नंबर है, काम हो गया. So the protocol which allows apps to communicate to each other, we call them APIs. So now you can answer this question. It can be integrated into any electronic device. The answer is yes. Laptops, computers, कुछ भी हो, mobile phones, tablets, कहीं पे भी आप use कर सकते हो. Online authentication using iris is possible. Yes, that is also possible. Finger, जहाँ पे भी अगर biological data stored है, then with help of that biological data, which can be fingerprint, आधार जैसे use करता है आपका, जब आप आधार बनवाने गए थे तो लिया था ना उन्होंने आपका iris scan. So it is possible. और iris scan जो है सबका unique है. So using iris scan, authentication can occur for much more secure things. कुछ ऐसी चीज़ें होंगी जहाँ पे बहुत ज़्यादा सिक्योरिटी चाहिए, तो वहाँ पे आपको iris scan से authenticate करना पड़ेगा. So yes, it is possible. Answer is both 1 and 2 here. This is not a relevant question anymore, but we should know about this also. जो 4G development है, that happened in many stages. जो 4G network है, 4G का जो standard है, that was announced by ITU, International Telecommunication Union, I think in 2007 or 8, I don't remember exactly which year. But the 4G technology took some time to be developed. We called it long-term evolution. But there was a problem in 4G network. 4G was supposed to provide you high-speed internet compared to 3G. But there was a problem in 4G network. That if you are on a phone call, that was the primary purpose of mobile network, कि आप बात कर सको. बट अब जो है, अगर आपको अब कोई बोले कि प्राइमरी पर्पस बात करना है, तो आप बोलोगे कि नहीं नहीं ये तो नहीं है. मुझे नहीं पता इसका प्राइमरी पर्पस क्या है. इसका प्राइमरी पर्पस ये है कि हम क्रिएटिव काम ना कर सकें, हम हमेशा बस इस फ़ोन में बिजी रहें. तो इसका प्राइमरी पर्पस एज ऑफ नाउ तो देखो यही है कि हर 10 सेकेंड में मेरे अंदर एक वो है कि नहीं मुझे देखना है, क्या पता दुनिया में कुछ हो गया हो, कुछ तो हो गया दुनिया में. एनीवेज, सो this network, 4G network, had a challenge that if you are on a phone call, then it did not know how to package your voice data. And at that point of time, if you want to explore internet, then that internet data together and used on the same 4G network. And 4G network was more efficient, provided you much better speed. But if you are on a phone call, then it did not know how to do them together. So your internet data used the 2G and 3G network, and your voice data used 4G network. So if you are on a phone call, your speed suffered. But then some companies figured it out, how to package them together and how to allow them to be transmitted on the same 4G network. That was called VoLT, Voice over Long-Term Evolution. In India, Jio was the first company to implement VoLT network in India. I remember this very specifically in 2016, I would say, or 15. There was a revolution that suddenly everybody was buying Geo SIMs. So first option, that LT is commonly marketed as 3G, VoLT is commonly marketed as advanced 3G. Answer is no, 4G. That 3G, it's not 3G, it's 4G. And LT is data only, VoLT is voice only. Both of them are both data and voice. The only thing is that LT does not know how to package them, voice data, internet data together. And VoLT can package them together. तो ये भी statement गलत है. Answer here is neither one nor two. So LTE is data plus voice. VoLTE is data plus voice. VoLT packages them together, allows them to be transmit with 4G network. LTE does not know how to do that. This was a question which was very much expected. In fact, I had hoped that there will be a question in mains examination also. अब इस साल डेटा प्रोटेक्शन एक्ट पे आ गया क्वेश्चन मेंस में. तो डेटा प्रोटेक्शन पे काफ़ी दिनों से बात चल रही है. ऑल दिस टेक कंपनीज, दे आर गैदरिंग सो मच ऑफ डेटा ऑफ इंडिविजुअल्स एंड दे शुड बी हेल्ड अकाउंटेबल हाउ दिस डेटा इज बीइंग यूज्ड और मिसयूज्ड बाय देम. So European Union took a lead here and they came up with set of principles, 7 principles, which needed to be legally implemented by all the tech companies operating in Europe. That पहले तो unnecessary data आप store नहीं करोगे, जितने टाइम तक डेटा स्टोर करने की ज़रूरत है उतने ही टाइम तक करोगे, डेटा सिक्योरिटी में इन्वेस्ट करोगे, बहुत सारी चीज़ें, लाइक वेरी गुड प्रिंसिपल्स. एंड यूरोपियन यूनियन टुक अ लीड विच वॉज फॉलोड बाई मेनी कंट्रीज इंक्लूडिंग इंडिया. एंड सो वी हैव डेटा प्रोटेक्शन एक्ट फॉर एग्जाम्पल राइट नाउ. सो यहाँ पे आंसर जो है वो सी है. ये फैक्चुअल क्वेश्चन है बट ये बहुत ही पॉपुलर फैक्ट अभी भी है और तब तो था ही. यूरोपियन यूनियन हैड अनाउंस्ड दिस आई थिंक इन 2017 or 18 and this question was asked by UPSC in 2019. There is a question on augmented reality and virtual reality. This is augmented reality, where with help of digital projections, I am enhancing my surrounding reality. Just right now, if I have to explain something, I have to show you, I'm showing these pictures. But what if I could use this part of this space with some sort of digital projections, with some app or something, and a 3D digital projection which can further enhance the appeal of the class. That is what augmented reality is. That I'm part of the reality. I am not some part of simulation. I know about where I am. I'm simply using digital projections to enhance that reality. Augmentation means enhancing. In virtual reality, often with help of some VR glasses, these specific devices, you become part of a simulation, a virtual simulation, and then you completely get immersed into that simulation. You lose your awareness about where you are, and your actions now have consequences in that virtual simulation. It started for video games, that many players all across the world can be part of one simulation and play a video game. But as of now, it has many applications. It has applications in healthcare, in education. In healthcare, for example, let's say a young surgeon is practicing a surgery in a virtual simulation on a patient, and then will actually do the real surgery in real life. In education, I can have this virtual classroom with help of VR glasses, some virtual classroom where we all are sitting here, same way, although we are not physically there, but we still are present, we still can talk to each other with help of our digital avatars. So that will be what virtual reality is, becoming very popular. And इसी का एक जो advanced version है, that a very large simulation where, so VR is often used. अभी कुछ कुछ malls में आप चले जाओगे तो वहाँ पे VR games का option मिल जाएगा. आप कोई VR glasses पहना देंगे, आप जाके video game खेलो, कुछ कार वार रेस कर रहे हो आप. But then some companies, and the most important company here is Facebook, thought that let's create a virtual world where you can do everything. That you make friends, do social networking in this virtual friend, play video games, buy some things. This virtual world will have its own set of rules, own set of regulations, buy property there, right? And this is called metaverse. In fact, Facebook is so bullish on metaverse that they have named their company, parent company, as Meta. That Meta owns Facebook, WhatsApp, Instagram, all of these companies. Thankfully, it has not become very popular. I often feel that this is a good news that metaverse has not become popular, because we already are isolating ourselves in this digital identity. We have more friends in digital domain rather than real friends in life. So metaverse is going to further isolate human beings, that you only are part of some simulation rather than part of real world. But this was asked by UPSC this year also. 2024 का जो एक prelims का question था, precisely यही था. Which one of the following word phrases is most appropriately used to denote an interoperable network of 3D virtual worlds that can be accessed simultaneously by millions of users who can exert property rights over virtual item? This is description of metaverse. I remember a few years ago, one of my friend asked me that metaverse में property खरीदते हैं. And I was like, I do not understand that economy. I will not do that. So anyways. Coming back to this question, AR, sorry? I do think that metaverse can actually explode in future. I actually do. I totally agree with you. There is a very good chance that metaverse will explode in future. I simply do not want it to happen. That was a personal opinion. But just because I do not want it to happen does not mean that it may or it may not happen. But I'm glad that it has not happened till now. Anyways, this next question is augmented reality versus that question which I was talking about. In AR, a simulated environment is created, physical world is completely shut out. In VR, images generated from a computer are projected onto real life objects or surrounding. ये exchange कर दिया बस दोनों statements को. AR allows individual to be present in the world, improve the experience. VR closes the world, transposes an individual, providing complete immersion experience. That is true. So first and second that have been exchanged, third and fourth, their correct answer here is B. There are really good virtual simulations these days. Virtual simulation glasses जो है Facebook का जो है Oculus Pro, that you can buy. I think it is I don't know, 60, 70,000, I don't remember the price. And there are really good simulations. There are simulations of Mount Everest. There are simulations of International Space Station, which you can be part of, right? There is another technology which is becoming very popular that is called mixed reality. In mixed reality, you are part of both worlds. So you are part of some simulation, but you have not lost the contact with the physical world, that you are aware about the physical world also. Or जो Apple का Vision Pro glass है, that is a very good example of that. That you are part of a simulation, but also you are part of the physical world. So that is called mixed reality. This was a very good question asked by UPSC. A digital signature, 3 statements, an electronic record that identifies the certifying authority issuing it, used to serve as a proof of identity of an individual to access information or internet, an electronic method of signing an electronic document and ensuring that the original content is unchanged. If you have not read about this topic properly, then you'll think that even first and second statements are correct, but they are not. There is something called digital signature certificate which can lead to at least first is true for that. But if I am using the word digital signature, it has a very simple meaning. That signature जब हम करते हैं documents का, तो उसका basis ये होता है that सबका signature जो है unique है, जो hard copy पे हम signature करते हैं. और बहुत सारे documents आजकल soft copy हैं, तो उसको हम कैसे ensure करें? So for that we use something called public key cryptography. That we create a public and private key. That public key is used to sign, but that private key is used to decipher. जैसे मान लो हमने digital sign कर दिया, किसी document को आपने अपना digital sign add किया, and this digital sign, if somebody has tampered with it, then for the person this document is meant for, can use its key to know that if it has been tampered with. So this is a type of encryption technique where the goal here is to understand that if a document has not been tampered with, right? So this is the purpose of digital signature, and that is why in this question the answer is 3 only, that it is an electronic method of signing. And it is not this electronic method of signing where you click a picture and add that picture on some PDF. It is not that. It essentially utilizes encryption and decryption technique using a mathematical way to ensure that your data is secure and your data has not been fuzzed. And there was a question on similar theme asked by UPSC in 2020 also, that they had directly now given this technology, public infrastructure, and you're supposed to know what it is. So public infrastructure में देखो क्या होता है, जैसे आप WhatsApp पे end-to-end encryption आप use करते हो, right? ये end-to-end encryption देखो कैसे काम करता है. I want to send some data to somebody, but I do not want any third party to know what that data is. तो उसके लिए हम use करते हैं these coding schemes to hide my data, and we call this encryption, right? हमने data को कुछ और तरीके से बदल दिया है, और जो तरीके से बदला है उसका जो लॉजिक है वो इतना आसान नहीं है डिसाइफर करना. वो लॉजिक डिसाइफर करने के लिए यू हैव टू सॉल्व अ मैथमेटिकल प्रॉब्लम, एंड इवन इफ यू हैव अ वेरी पावरफुल कंप्यूटर देन आइडियली यू शुड नॉट बी एबल टू सॉल्व दैट मैथमेटिकल प्रॉब्लम. जैसे एक सिंपल सा क्वेश्चन देखो, यूपीएससी सीसेट पेपर 2 में क्वेश्चन पूछती है वो वाले जहाँ पे some number, some letters are given, and in that coding scheme some number means this letter, this number means this letter, and then you are supposed to know the meaning of the third one. So they are using some sort of logic there, and based on they are expecting you to decipher that logic in less than 1.5 minute and answer that question. So clearly जो encryption scheme UPSC आपसे expect कर रही है decipher करने के लिए बहुत strong नहीं है कि आप उसको 1 मिनट में 1.25 मिनट में आप उसको decipher कर लो. But the encryption schemes which I would ideally want in communication systems, in data storage system, should be very strong. That even with very powerful computers, I should not be able to make sense of the data. This is where this public infrastructure comes into place. And there are many ways to do that. जो सबसे simple तरीका है और जो सबसे effective तरीका है वो है one mathematical problem multiplying two very large prime number. So if you multiply prime, remember prime numbers, only 2 factors, number 1 and number itself. So think of P and Q as 2 prime numbers, multiply them, you get R. For encryption, you use this technique. But for decryption, R is given to you and you are supposed to know its prime factor. The same problem in the opposite direction becomes so difficult that it cannot be solved even with very powerful supercomputer. So if two people want to send data to each other, जैसे मान लो कि there is a person A, there is a person B. A and B want to ensure that whatever they are communicating, some C should not be able to know what that data is. A and B have to agree on some encryption and decryption key, and they also have to ensure that this decryption key should not be easily decipherable by C. How they will work, how they will use this? A will use its encryption key and transmit it on a public network. कि मान लो कोई A है, A ने क्या करा, जो encryption की है पूरे नेटवर्क पे सबके साथ शेयर कर दिया है. अब जिसको भी A को डेटा भेजना है, they can use this encryption key, use this encoding scheme, send that data to A. Now the thing is that A has a private key now, and A did not share that private key with anybody. A kept that private key located to its device, and with help of that private key, A can decipher that specific data. And if C wants to know what that data is, then C has to solve for that private key. And that private key ka logic kya hai? That there is a very large number given, you have to find its prime factor. C, even if brings the most powerful supercomputer, will not be able to solve that in a real time frame. And since you share that encryption key on a public network, this technology is called public key distribution or public key infrastructure or public key cryptography. All three of them are same thing. तो यहाँ पे जो आंसर था बड़ा सिंपल था, A, digital security infrastructure, and the same public key cryptography is also used in digital signature to check for encryption and decryption key, that if it has not been tampered with. ये easy question है. Wearable devices, what they can achieve, location identification of a person. If that wearable device has a GPS electronic device, then of course I can locate it. Sleep monitoring of a person, yes, there are many devices which monitor sleep. Assisting the hearing impaired person, ये बहुत पुरानी चीज़ है, ये तो नया भी नहीं है. So third is also true, answer is all three of them are correct. This is a controversial question, application of AI. यहाँ पे UPSC के हिसाब से जो आंसर है वो आपका 2 is clearly true, 3 is clearly true, 2 is wrong. So UPSC considered 2 to be wrong, and they gave the answer as 1, 3 and 4. Bringing down electricity consumption in industrial unit, disease diagnosis, text to speech conversion. They did not consider that create meaningful short stories and songs to be correct. And they did not consider that 5 wireless transmission of electrical energy to be correct. I have a problem with this. UPSC का जो official answer की था, that was 2 was wrong. I can still appreciate it a little bit, although even that statement is subjective. जैसे जो create meaningful short stories and songs, ये meaningful word जो है बहुत ही subjective word है. Meaningful का कोई meaning नहीं है as such. Meaningful का जो meaning है वो इस बात पे decide होगा कि पढ़ने वाला क्या meaning निकाल रहा है. There can be some story which can be meaningless for somebody, but there can be story which can have like grand meaning for somebody else. So these are subjective words, and asking the subjective words in objective question does not make sense. But I get what they are trying to ask. All of the LLMs we are using, ये LLM का capability होगा, large language model का capability होगा. Even today, ये तो 2020 का question है, and question was also asking based on present state of development. तो 2020 में ChatGPT का जो 4th version है, it was not there. GPT-1 was there, 2 was there, there was no 3, there was no 3.5, there was no 4. So that question was based on the earlier capability of large language models. They were capable of translation, analyzing large set of data, hence in disease diagnosis. Meaningful का मतलब होता है that you have done something original, and because जो GPT है, जो भी ऐसे tools हैं, they have been trained upon some data, and based on that training data they have created something. So is it original? But then you ask the same question for all the authors also, that if an author has written a story, then that also may be inspiration due to many things that author has witnessed in his or her life, learned from things, learned from life. So we are saying that GPT, whatever it did, was based on training, so it has no meaning. But author did that, it has meaning. So my point here is that I understand why they thought this to be wrong. जैसे if there अभी भी अगर मान लो एक statement ऐसे आ जाए composing original song AI का capability, तो I will consider that to be wrong. Original song जो है हम ये मानते हैं that it is something which emerged out of this human creativity, right? But if it is about song lyrics, then AI can be better than all Tony Kakkars of the world. मतलब ऐसे समझ लो आप. So or Badshah of the world. So you can think of it like this, that that second statement जो है, that second statement, if it is even today it is composing original music, then I will consider wrong. But in this current form, this is controversial. But then I also have this problem with fifth option. First option, that bringing down electricity consumption in industrial unit. Let's understand the logic of it. How much power is required? Optimization of power requirement is a mathematical problem. And AI can provide a solution, a better solution, where I can optimize my power requirement. So first makes sense. But so is fifth, that wireless transmission of electrical energy, of course it is possible. Wireless transmission of electrical energy happens with help of magnetic induction, electromagnetic induction. Do I find a better way to do electromagnetic induction? Because electromagnetic induction, also doing this, is a engineering problem. It happens, we already use it. आपने wireless charging पढ़ा होगा, दो मोबाइल फोन पास में आ जाए, दूसरा मोबाइल फोन चार्ज हो रहा है. So this is a mathematical engineering problem. Can I find a better solution with help of AI calculation? Why not? If I can do the first, I can also do the fifth. But somehow UPSC thought that fifth is not correct. I can still appreciate second, that what they're trying to say, why they considered it wrong, but I cannot understand why they thought fifth to be wrong. So I cannot explain UPSC's official answer key. I can explain two, and even then I will mention that this is highly subjective, and in objective question, such subjective interpretation should not be asked. Anyways, let's go to the next question. We have done that. Blockchains. Blockchains are distributed ledger. We have learned that blockchains are distributed ledger. Once records are added here, those records cannot be manipulated. And the best part of blockchains are that they are not stored, or nobody, one entity can claim the ownership of this entire record. They're not stored at one central server. They are stored across many computers, geographically distributed. We call them nodes. If you want, you can also be part of a node of a blockchain, such as Bitcoin blockchain or Ethereum blockchain. So the statement is saying that it is a public ledger that everyone can inspect, but which no single entity controls, single user controls. That actually is the definition of blockchain technology. The structure and design of blockchain is such that all the data in it are about cryptocurrency only. So blockchain was designed for cryptocurrency by this person called Satoshi Nakamoto. We do not know who that person is. He designed blockchain for this first cryptocurrency of the world we call Bitcoin. And yes, Bitcoin blockchain even today is only can be used for Bitcoin purpose only. But the thing is that people have now found many alternate way to design blockchains, and those blockchains can have applications in many more domains: financial, data, smart contracts, Web 3.0. So many different applications are there. So that second statement cannot be considered to be correct. And applications that depend on the basic features of blockchain can be developed without anybody's permission. That is also true. If a blockchain allows you to develop an app, then you do not need anybody's permission, because remember, nobody owns it. Even if somebody has written a code, to run that code, they need geographically distributed nodes of computers. And you cannot own all of them. The moment you own all of them, then essentially that becomes a data center. And blockchain is not a data center, centralized data center. So that third statement is also correct. So there are certain apps called decentralized apps. And decentralized apps are supported not by some centralized cloud server, but by decentralized network of computers. And they do not need anybody's permission to be developed. So answer here is 1 and 3 only. Second option can be eliminated quite easily. Third was a little bit difficult, but even third statement is true here.
A strong hold over current affairs is your way to succeed in the UPSC Civil Services examination. Brace yourself and get detailed coverage of relevant news both nationally and internationally. With the monthly current affairs revision program starting from 26th October at 5 p.m. Enroll today and elevate your current affairs knowledge with comprehensive coverage of current affairs compiled through relevant sources such as the Hindu Indian Express, Economic Times, PIB Yojana, and others. Expert support and personalized mentoring. Comprehensive assessment through MCQs and mains-oriented questions.
This is very easy. Drones के बारे में पूछा है कि ड्रोन से हम क्या क्या कर सकते हैं. Can we spray pesticides on a crop field with drone? That's very famous. Inspecting the craters of active volcano. That's also very simple. Collecting breath samples from spouting whales for DNA analysis. This actually was done like 1 or 2 years ago before this question was asked in 2020. And because of that statement was given. But yes, that can also be done. A very small drone, whale will ignore it like we ignore flies. But that drone can collect sample and we can analyze DNA of whales and we can understand how DNA is how whale is connected to other mammals of the land, because remember whale is a mammal, right? So answer here is 1, 2 and 3. This was a very easy question. D is the correct answer. This was a question again due to COVID-19. Government of India made this app called Arogya Setu app. The purpose of Arogya Setu app was, have you guys used it? The purpose here was that with this app, it can transfer data with help of Bluetooth from each other, and it uses GPS also to locate. So if let's say for example, you are in contact with a person who is infected with COVID for very long, you visited a hospital, let's say that hospital has some COVID patients and you spend some time here, then there is a chance that your mobile will get this alert by that person's Bluetooth. And you will come into this orange category. Aapka jo Arogya Setu, it will show you orange. And if you are infected, then in any case, this was a legal mandate that the moment a lab finds you COVID positive, then they have to alert to the government. Aapka number Arogya Setu पे alert है, वहाँ पे number जो lab ने उन्होंने share कर रखा है, आप पे वो signal red आ जाएगा that you are infected with COVID. But your location data now will be shared with other devices. Your personal data will not be shared. Your name, your phone number, nothing will be shared. But in your area, जैसे मान लो आपको अभी COVID नहीं है, in your area you can see that in 10 kilometer square area, how many people are infected. So that was the purpose to understand about infection, to do contact tracing, all of those things Arogya Setu was developed. Now some people said that Arogya Setu is a problematic thing because it is sharing personal data, it can be exploited, etc., etc. Ministry of Electronics and IT said that if you feel it so, then we are going to make it open source platform, which essentially means that the entire source code of Arogya Setu was made in public. And then Government of India said that if you feel that there is some problem somewhere, then make suggestions. No problem was found. This was simply an allegation. And then Government of India has done this not just for Arogya Setu but for other platforms also. And the answer here is all 4 of them. CoWIN was another platform which was used for vaccination now. कि आप वैक्सीन का डेट ले सकते हो, देख सकते हो कहाँ पे स्लॉट्स अवेलेबल हैं. DigiLocker is where we store our documents. Diksha is also a portal, I guess, used for education or degrees, something like that. All 4 of them are example of open source digital platform, which means that you can see source code. A truly open source also allows you to make changes. यहाँ पे बहुत limitation है कि आप क्या change कर सकते हो, क्या नहीं कर सकते हो, but you can clearly see the open source of the code of these platforms. And I was very surprised to see that this question was actually asked by one of the editorials in Indian Express. And when I saw this question, I remembered reading this editorial. This editorial was advocating that apart from these 4, which already are, these 4 were clearly mentioned. I have given the link here. You can go and click on this link. Exactly these 4 were mentioned. And the editorial was advocating that maybe it can be done for other services also. That was the main gist of the editorial. And somebody was reading that editorial and asked these 4 options and asked a question in prelims. Answer is D here. सॉरी, विकिपीडिया सोर्स कोड आप देख सकते हो, चेंजेस कर सकते हो. वेब 3.0. सो इंटरनेट का जो इवोल्यूशन है, वी थिंक ऑफ दिस इनटू 3 generations. The first generation of internet was certain web pages are there. You open these web pages, gather some information. Most of that web page is essentially document texts. There are some images, maybe some videos also. You switch it, you close it down. They are not collecting any of your data. आपको कुछ login credential, कोई data collection है नहीं. And you cannot make any changes to those web pages. जो web page है, वो web page है. उसमें आप नहीं change कर सकते. जो change करेगा वो web developer करेगा. So this which continued till 2000s, we call it Web 1.0. But around 2002, 2003, some remarkable changes happened. That with help of cloud platforms, with help of advancements in programming languages, people were, or tech companies were able to develop certain portals where two things happened. That not only you are accessing information from internet, even your information is being accessed by the ISP or browser. So they are now gathering, storing your information, your email ID, your phone number, etc., your browsing history. All of those things are being stored by your ISP, by web browser, the website you have visited. They're trying to make a profile out of you. So first thing. And second thing, on certain portals, particularly on social media, you can go and make changes on the web page. So for example, when you post a tweet on nowx.com, then that tweet essentially is new content on that Twitter. That was not possible in Web 1.0. But that first change is more why we use this Web 2.0, that even this is no longer one-way access of information, this becomes two-way access of information. And that enabled certain tech companies to become so powerful, such as Google or Amazon or Microsoft or Facebook. These companies are more powerful than many governments of the world. These companies invested into giant data centers, cloud servers, which enabled them to achieve this. But that led to this serious criticism that these tech companies are storing data of people, and how they are using this data, people do not have control over that. So this is where some technocrats advocated for evolution of internet into what we call Web 3.0. That Web 3.0 is not, by the way, one technology. Web 3.0 essentially is certain set of principles, and to implement those set of principles we need technology, that goes without saying. But Web 3.0 in itself is not a technology. Web 3.0 का जो main principle जो है वो ये है that people are at the center of internet. Rather than going for all these centralized data servers and data centers, we should use blockchain-based networks. On these blockchain-based networks, people should have control over their own data. That if they want to share data with somebody, they should be allowed. If they do not want, they shouldn't do that. If some of the data they feel is sellable and they want to sell it, then they should do that. It shouldn't occur due to some other companies. The transactions which happen should occur without some third-party intermediaries. We call this smart contract. Right. So all of these are fundamental principles behind Web 3.0. It is here. There are some apps which is based on Web 3.0. There are some browsers which is based on Web 3.0. The only problem is that they're not interactive as such right now. They're not also very smooth, not very fast, so it is not taking up. Will this how internet evolve? We simply do not know. Maybe it will not evolve like this. That is why I said that Web 3.0 is, I'm only saying it that it is a set of principles and we need technologies to fulfill those principles. The first statement is that it enables people to control their own data. That is the foundation of Web 3.0. There can be blockchain-based social network. That will be the most important technology of Web 3.0. If you want to reduce your dependence upon cloud servers and data centers, then blockchain is the way to go. Is operated by users collectively rather than a corporation. That is also a definition of Web 3.0. So answer is here is 1, 2 and 3. All three of them are correct statement. This was in news a lot, this has been in news a lot, and because of that UPSC asked a question from this topic. सॉफ्टवेयर एज अ सर्विस का देखो क्या मतलब है. आज से आप 20 साल पहले अगर आपको एक सॉफ्टवेयर लेना होता तो आप एक जाते, एक सीडी मिलता आपको, एक फ्लॉपी डिस्क मिलता, उसको आप अपने कंप्यूटर पे, डेस्कटॉप पे लेके आते, इंस्टॉल करते, वो काम करता, दैट्स इट. उसके बाद कोई सपोर्ट नहीं है. आपका कंप्यूटर क्रैश हो गया, कुछ इंपॉर्टेंट डेटा था आपने स्टोर नहीं किया, कोई फर्क नहीं पड़ता. यू डू नॉट हैव एनी सपोर्ट. बट विथ दिस वेब 2.0, विथ ऑल दिस cloud based servers, now a lot of things happen. First, you can when you are buying a software, you do not have to go to some shop. You can go to a website, you can make payment, install software from there itself. But many of these softwares now provide you cloud support, which essentially mean that they provide you data storage. In some cases, they also can provide you data processing also. Right? जैसे मान लो आपने Microsoft Word है, कुछ भी है, ईमेल है, आप ईमेल किसी भी आप डिवाइस से ओपन कर सकते हो. आई कैन लॉग इन माई ईमेल फ्रॉम दिस फोन बट ऑल्सो फ्रॉम माई लैपटॉप. आई कैन ओपन अ Microsoft Word document here in this mobile phone if I have stored even though on my computer, but I made a backup on the cloud. So that cloud-based support to run operations, to run software, to make things easier for users is called software as a service. And if you want, you can also customize things. जैसे मान लो Microsoft Office में मुझे एक specific मेरा जो requirement है as an individual, मुझे पूरा ऑफिस नहीं चाहिए, मुझे Microsoft Word चाहिए, Excel चाहिए, PPT चाहिए. I can do that. मुझे बहुत ज़्यादा स्पेस की ज़रूरत नहीं है क्लाउड पे. मुझे हर मंथ मुझे मान लो 100 MB या 1 GB की ज़रूरत है, मैंने खाली उसका सब्सक्रिप्शन लिया है. सो आई कैन डू सच कस्टमाइजेशंस. दिस कस्टमाइजेशंस आर अवेलेबल बिकॉज ऑफ दैट क्लाउड बेस्ड support. So SaaS buyers can customize the user interface and can change data fields. That is true. SaaS users can access their data through their mobile devices. That is true. Outlook, Hotmail, Yahoo, these are the first SaaS platforms. But as of now, Outlook still is quite popular though in offices. Not that popular. Hotmail is also not that popular. Yahoo Mail is also not that popular. But the thing is that these are very good example of first set of SaaS platforms which was provided. So we are essentially talking about cloud-based software model, which is software as a service. So answer here is D, 1, 2 and 3. Quantum computing is a very difficult topic, but UPSC जो सबसे simplest question possible हो सकता है उन्होंने पूछ रखा है कि what is qubit? Qubit is a term which is used in case of quantum computing. In normal computers, we use these two binary states, which is either 0 or 1. So you store your data in binary, you process your data in binary. Binary को डिज़ाइन करना आसान है. I can have transistor switched on, which is 1, transistor switched off, that is 0. So उसको डिज़ाइन करना आसान है. In quantum mechanics, you can find something called quantum superposition. Quantum superposition means that both 0 and 1 can exist simultaneously. When you describe nature at very smaller scale, such as electrons or protons or atoms and nuclei, then you come across this property called superposition. So a state of electron can be represented with a state of superposition. And if that one's of state, I think 0, another to be 1, then I can have both of them together. And with help of qubits, now I can do calculations, and those calculations become quite faster. So qubit is the basic unit which we use in case of quantum computing. So this was the simplest question possible, and they did ask that. Next time when they will ask this, it will not be that simple. But even then it will not be that difficult. Maybe it will be 2 statement or 3 statement question, or maybe there will be a question about National Quantum Mission, or maybe there will be a question in mains also from this topic. Which of the above are considered short range devices technologies? Right? अब देखो, wireless local area network, radio frequency identification, CCTV. All 3 of them are short range. देखो devices word भी use किया है. तो CCTV is a short range device. So CCTV, it can capture, you know, what is CCTV? That is a short range device. RFID is a short range technology that आपकी गाड़ी में जो फास्टैग लगा हुआ है वो जब आप टोल बूथ पे जाओगे तभी वो रीड कर पाएगा उसको. बहुत दूर अगर टोल बूथ से, ऐसा नहीं है कि आप बहुत दूर खड़े हो टोल बूथ ने रीड कर लिया, तो उसका अपना रेंज होगा. ऑल्दो RFID का जो रेंज है, it differs depending upon which frequency is being used, but even when you are using very low frequency, the range will be not more than few tens of meter or maybe 100. I don't think it is even 100 meter. Maximum range can be few tens of meter. That's it. So, and wireless जो WLANs है, yes, that is also true. जो wireless local area network है, that is also an example of short range technology. Wi-Fi is one of the best example. So you have all three of them are correct here. Answer is D here, 1, 2 and 3. I do not consider this topic to be important anymore. Although I do consider blockchain technology to remain important in coming years. One of the application of blockchain technology is non-fungible token. Token का मतलब होता है that you essentially are representing something else with some sort of token. राइट कि आप जाते हो कोई शॉप में कुछ खरीदने तो आपको एक टोकन मिल जाता है, टोकन लेके आप जाते हो वो सामान मिल जाता है आपको. सो टोकन इज अ रिप्रेजेंटेटिव ऑफ समथिंग. देयर आर सर्टेन टोकन्स विच यू कैन एक्सचेंज ऑल द टाइम एट इक्विवेलेंट प्राइस. राइट, देयर आर सर्टेन टोकन्स विच यू कैन नॉट. जैसे करेंसी इज वन ऑफ द बेस्ट एग्जाम्पल ऑफ फंजिबल टोकन. That I have 100 rupees, I can ask 250 rupees from you. You will not have any problem in doing that. But there are things which cannot be valued by everybody at the same value. जैसे मैंने पेंटिंग कुछ खरीदा 1000 रुपये का और मैं आपको बोलूँ कि मुझे 1000 रुपये दे दो, अब बोलोगे मैं क्यों दूँ 1000 रुपये. I will not give you any rupees, maybe I will give you 1 rupee. And it's totally fine, you can totally say that. So this is the meaning of the word non-fungible. So non-fungible token became very popular for digital piece of art. that जो digital artists हैं उनके साथ सबसे बड़ा problem क्या है, that उनके art को copy करना बहुत आसान है. We just have to do control C control V. Physical art copy को copy करना मुश्किल है, कर सकते हो, but everybody knows what is where the physical copy is, artist का signature है, बहुत सारे तरीके हैं. तो non-fungible token started as a some sort of ownership of digital piece of art to digital artists. That there is an art का देखो कोई definition है नहीं, art can be anything. So it started with digital images, memes, etc., etc. Somebody designed a NFT on blockchain, and that NFT now can be sold to somebody else by the owner of that NFT. But the only thing is that you cannot sell it every time at the same time. जैसे मान लो मैंने कोई NFT खरीदा कुछ एक इथेरियम में, I cannot, इथेरियम is a cryptocurrency, एक इथेरियम में I cannot sell the same NFT in one ethereum because other people are not valuing it at the same value. So it started for digital piece of art, but later it was also used for physical objects. कि कोई land piece है, उसका हमने एक token create किया है और हमने store कर दिया blockchain पे. That token is a proof that I own that piece of land. And मान लो कि अगर इसको government authenticate कर दे, कि government of India ने accept कर लिया कि there is this government agency, आपने टोकन बनाया, वो गवर्नमेंट एजेंसी अप्रूव करेगी कि ये जो टोकन है वो सही में करेक्ट है नहीं, आप ही इसके ओनर तो नहीं हो या कोई और तो नहीं है. मान लो गवर्नमेंट ऑफ कोई एजेंसी है, उन्होंने कर दिया, ऑथेंटिकेट कर दिया, इट इज स्टोर्ड ऑन अ ब्लॉकचेन. नाउ आई कैन सेल दिस टोकन टू समबडी एल्स, ओनरशिप ऑटोमेटिकली विल बी ट्रांसफर्ड. सो इट स्टार्टेड फॉर डिजिटल ऑब्जेक्ट बट इट वॉज यूज्ड ऑल्सो फॉर फिजिकल ऑब्जेक्ट. So the first option is that they enable the digital representation of physical asset. That is true. Although it did not start for that. It started for digital piece of art. They are unique cryptographic tokens that exist on blockchain. That is also true. They can be traded or exchanged at equivalency and therefore can be used as a medium of commercial transaction. That is the exact opposite to the word non-fungible. तो ये question इतना simple है that if I know the meaning of the word non-fungible, then I know that third statement is wrong, and if I know third to be wrong, I know my answer is 1 and 2. So science and tech भले prima facie थोड़ा difficult topic लगता हो, but questions जो होंगे बड़े ही ज़्यादा attemptable questions होंगे, maximum questions जो है attemptable होंगे, और थोड़ा सा logic लगाना है आपको, basic understanding होना चाहिए, and you can get the answer correct in most of the questions. There was a question on sensor. There is a sensor called accelerometer. अब देखो कुछ कुछ सेंसर हम एज आइसोलेशन डिवाइस यूज करते हैं. थर्मामीटर इज अ सेंसर जो आपका बॉडी टेम्परेचर बताता है, राइट. बैरोमीटर इज अ सेंसर विच मेजर्स प्रेशर. बट मान लो मेरे रूम में एक ये भी सेंसर हो सकता है, जो मैं एग्जांपल दे रहा था IoT में. देयर इज अ थर्मल सेंसर विच इज मेजरिंग बॉडी हीट. इफ देर इज नो पर्सन, then it sends a signal to AC becomes switched off. This is the idea behind Internet of Things. But those sensors can be used for multiple other purposes. There is a sensor at Karol Bagh Metro कि escalator रुका हुआ है, जैसे यहाँ पास में जाओगे वो चलना शुरू कर देगा. Clearly there is a sensor which is measuring your mass, and based on that the escalator starts moving. अगर बिना मतलब का चलते रहे तो बिना मतलब का loss of energy है. escalator चलाने के लिए एनर्जी चाहिए ना, इलेक्ट्रिकल एनर्जी चाहिए. अब एक इंसान दिन भर खड़े होकर स्विच ऑन स्विच ऑफ करते रहे, दैट इज ऑल्सो नॉट अ गुड सेटअप. सो हमने क्या किया, सेंसर लगा दिया. टैप पे हमने थर्मल सेंसर लगा रखा है. पुट माई हैंड बिलो अ टैप, वाटर ऑटोमेटिकली कम्स आउट, देयर इज नो स्विच देयर. सो देयर आर मेनी टाइप ऑफ सेंसर्स विच आर इंटीग्रेटेड इन आवर लाइफ, and these sensors collect some data, and based on that data collection they send a signal to some electronics that this instruction should be executed. One of that sensor is accelerometer. Thermometer measures temperature, barometers pressure. What will accelerometer measure? Acceleration. So the statements are detection of car crash collision which results in deployment of airbag almost instantaneously. That is actually done by accelerometer. The answer is correct. सडनली आपका अगर कार रुक रहा है तो फिर वो जो है एक तरीके का एक्सेलरेशन ही है. उसको डिसेलरेशन हम बोलते हैं, बट है तो एक तरीके का एक्सेलरेशन ही. डिटेक्शन ऑफ एक्सीडेंटल फ्री फॉल ऑफ लैपटॉप टुवर्ड्स द ग्राउंड विद इन विच रिजल्ट्स इन इमीडिएट टर्निंग ऑफ द हार्ड ड्राइव. दैट इज ऑल्सो टाइप ऑफ एक्सेलरेशन. इफ हार्ड ड्राइव इज फॉलिंग डाउन, इट्स स्पीड इज इंक्रीजिंग, it is an acceleration. And yes, there are certain hard drives equipped with accelerometer which can switch off things. That does not mean that they can avoid physical damage. The only thing is that whatever data was saved here, if it is not physically damaged, it can be recovered easily. So yes, accelerometer is used there also. And this, doing this portrait landscape mode also, we use two sensors actually. The first sensor is accelerometer. The second sensor is gyroscope. Accelerometer measures the remember, if I am changing my orientation, then even this is acceleration. So accelerometer measures this change in orientation. Gyroscope maintains the stability, that the smooth transition from landscape to portrait, that occurs with help of gyroscope. But this has happened, this is done by accelerometer. So answer is all three of them. Answer here is C, all three. Okay? This we discussed. Can I extend like this class? I do not need to take this extra like 15-20 मिनट और हम कोशिश करते हैं कि जल्दी जल्दी इसको ख़त्म करेंगे. INS अस्त्रधारिणी, दिस वाज अ क्वेश्चन इंस्पायर्ड फ्रॉम करंट अफेयर्स. ये तब करंट अफेयर्स, डिफेंस के क्वेश्चन करंट अफेयर्स से ही आएंगे, तब ये बहुत ज़्यादा न्यूज़ में था. अ न्यू टाइप ऑफ शिप कॉल्ड ऑक्सिलियरी शिप्स. ये जो शिप है इसका पर्पस था, दिस वाज अ टॉर्पीडो लॉन्च एंड रिकवरी वेसल. तो इसको हम ट्रेनिंग के टाइम पे देखो हम क्या यूज़ करते हैं. वी लॉन्च टॉर्पीडो इज दिस अंडर वाटर मिसाइल, कि अगर मुझे किसी अंडर वाटर सबमरीन को डिस्ट्रॉय करना हो या किसी सबमरीन को ऊपर सरफेस पे शिप को डिस्ट्रॉय करना हो, दे लॉन्च टॉर्पीडोज. तो INS अस्त्रधारिणी का गोल ये है दैट ड्यूरिंग ट्रेनिंग इट कैन लॉन्च अ टॉर्पीडो बट इट कैन रिकवर इट ऑल्सो. इट कैन डिप्लॉय मैकेनिज्म्स टू रिकवर दैट टॉर्पीडो, राइट. सो ये मिलिट्री एक्सरसाइज में हमारे लिए बहुत फ़ायदेमंद है. एक्चुअल वॉर में, बिकॉज टॉर्पीडो को हम लॉन्च करेंगे, उसका ट्रेजेक्ट्री देखेंगे, कैसे मूव करता है, कितने स्पीड से एक प्लेस पे हिट करना है, कैसे हिट करना चाहिए. फॉर दैट दिस रिकवरी पार्ट इज ऑल्सो इंपॉर्टेंट. तो ये जो पूरा वर्ड क्या है, टॉर्पीडो लॉन्च एंड रिकवरी वेसल. तो लॉन्च तो हम बहुत जगह से कर सकते हैं, बट दैट रिकवरी पार्ट इज इंपॉर्टेंट टू अंडरस्टैंड द मैकेनिज्म ऑफ फ्लाइट ऑफ टॉर्पीडो. एंड दैट इज वाई INS अस्त्रधारिणी वाज बिल्ट. This knowledge allows us to actually use torpedo during a naval warfare, right? So यहाँ पे answer C है. I do not expect you to know that, simply because this was in news a lot, by the way, in 2016, and UPSC asked a question on this topic. Third is again in news. So there are many air defense systems across the world. Iron Dome system of Israel has been in news continuously because of Israel multi-front war. As of now, it is fighting. But THAAD is again another very powerful air defense system of America, which they have deployed also at the border of South Korea and North Korea. At border of South Korea, North Korea, साउथ कोरिया का जो डिफेंस इंडिया साउथ कोरिया एंड यूएस के अपने खुद के डिफेंस पैक्ट्स हैं. So THAAD is deployed there, that if there is a North Korean missile being fired, then THAAD can intercept and neutralize it. But this again is in news. At that point of time, it was in news because of that North Korea, South Korea, हमेशा होते रहता है वहाँ पे भी, and some confrontation was going on, and this was in news. This is in news again because Iran has very powerful missiles. So America has deployed THAAD to Israel. So now Israel has this David Sling, Iron Dome, THAAD, all of these are air defense systems to protect cities in Israel. So answer here is an American anti-missile system. Fractional orbital bombardment system, देखो ये क्या होता है. आपको एक बहुत बड़े टारगेट पे आपको हिट करना हो बहुत दूर, यू यूज अ इंटरकॉन्टिनेंटल बैलिस्टिक मिसाइल. इंटरकॉन्टिनेंटल बैलिस्टिक मिसाइल का प्रॉब्लम क्या होगा कि जितना ज़्यादा रेंज होगा ये उतने ही हाइट पे जाएगा. सो इट ऑफन गोज मोर देन 1000 किलोमीटर हाइट. अब यहाँ पे अगर कोई रडार है, ये इसको आराम से डिटेक्ट कर लेगा बहुत पहले फ्लाइट में, एंड इट कैन न्यूट्रलाइज दैट. सो व्हेन कोल्ड वॉर वाज गोइंग ऑन between US and USSR thought of this technology called fractional orbital bombardment system. कि हम क्या करेंगे इस सिस्टम में, we will launch my missile in a very small orbit around Earth, about 100-150 kilometer. तो हमने रॉकेट लॉन्च किया, मिसाइल लॉन्च किया, but I achieved a very small orbit, about 100-150 kilometer. And now this is moving not because of its fuel, it is moving because of Earth's gravitational force. But it does have lot of fuel. Just before hitting the target, I deorbit it. So before hitting, I do not have to complete this orbit. Before hitting the target, हमने क्या किया? I used my fuel and I deorbited and I hit the target. इसका benefit ये है that this interception will happen quite late. And you can hit your target without alerting enemy's radar system. So this technology is called fractional orbital bombardment system, which was designed by USSR in 1970s. But why this was asked in 2021 or 2022? Because in the same year in January, there was a news from China that China has tested its hypersonic missile using this technology, fractional orbital bombardment system. And because of that, this topic was in news. So यहाँ पे जो answer है, C is the correct answer. A missile is put into stable orbit around the earth and deorbits over a target on the earth. अगर आपने डिटेल में नहीं पढ़ा था, अगर आपने खाली हाइपरसोनिक मिसाइल पढ़ के कुछ छोड़ दिया था, तो आप A मार्क करके आ जाओगे. But read the complete A. A hypersonic missile to destroy an asteroid, not at all. Fractional orbital bombardment system is to hit a target on earth while flying at very low altitude in a stable orbit. अभी आई गया, नेक्स्ट क्वेश्चन आई गया, वही है. बैलिस्टिक मिसाइल का मतलब ये होता है, बैलिस्टिक मिसाइल इज लाइक अ थ्रोइंग अ बॉल, राइट, अ बॉल लाइक ट्रेजेक्ट्री, अ बैलिस्टिक ट्रेजेक्ट्री. अ बॉल लाइक ट्रेजेक्ट्री क्या होगा? एक पैराबोलिक पाथ होगा कि अगर मुझे एक बॉल में थ्रो करूं तो इसका जो भी स्पीड, जो भी डायरेक्शन मुझे देना हो मैंने दे दिया. इसके बाद जो इसका पाथ है, आई एम नॉट डिसाइडिंग दैट. इसके बाद जो पाथ होगा दैट विल बी गाइडेड बाय ग्रेविटी. सो बैलिस्टिक मिसाइल इज लॉन्च्ड विथ हेल्प ऑफ अ रॉकेट. Why rocket? Rocket carries both fuel and oxidizer together. Why rocket? Because ballistic missile goes up the atmosphere, often crosses atmosphere. Outside atmosphere there is no oxygen, so you have to carry oxygen also. It goes outside. Rocket के अपने कुछ कुछ stages होंगे. मान लो अग्नि 5 मिसाइल है, जो कि बैलिस्टिक मिसाइल है, इंटरकॉन्टिनेंटल बैलिस्टिक मिसाइल है. 3 स्टेज का रॉकेट है. पहला स्टेज ने एक्सेलरेट किया, दूसरे ने किया, तीसरे ने किया. तो इसके बाद जब मेरा जो मिसाइल पार्ट है, वंस इट इज रिमूव्ड, अब इसका पाथ रॉकेट गाइड नहीं कर रहा है. अब जो इसका पाथ है दैट इज बीइंग गाइडेड बाय अर्थ्स ग्रेविटेशनल फोर्स. सो दिस बैलिस्टिक ट्रेजेक्टरी गाइडेड बाय अर्थ्स ग्रेविटी, हेंस द वर्ड बैलिस्टिक मिसाइल. इसका फायदा ये होता है दैट सिंस इट फ्लाइज आउटसाइड एटमॉस्फेयर, इट कैन गेन वेरी हाई स्पीड, 5 times, 10 times, 15 times faster than speed of sound, which we use a word Mach number. So it can use Mach 10, Mach 15, and it can hit a target far away in a very short span of time. But nuksan kya hota hai? That since it is coming from such high altitude, it can be intercepted, which we saw in the previous slide. This is where the second missile comes into place, cruise missile. That a cruise missile flies within atmosphere. And within atmosphere, you do not have to carry oxygen. So it does not have to be a rocket engine. We use what we call jet engine, turbojet engine. To be more precise, a turbojet engine. जैसे ब्रह्मोस मिसाइल जो है वो क्रूज मिसाइल है. क्रूज मिसाइल बैलिस्टिक मिसाइल जितना फास्ट नहीं उड़ सकता, बिकॉज ये एटमॉस्फेयर के अंदर इसको उड़ना है. So cruise missiles, many of the cruise missiles are subsonic, less than Mach 1. Some of them are supersonic, such as BrahMos. BrahMos is a supersonic. And some countries also have hypersonic. Hypersonic means more than 5 times speed of sound. Jase America has one, Russia has one. Iran also claims to have a hypersonic missile. So now you can answer this question that ballistic missiles are jet propelled at subsonic speed throughout their flight. सब कुछ गलत है. Ballistic missiles are not jet propelled. Jet engine does not carry oxygen. Ballistic missiles are rocket propelled, which carries both fuel and oxidizer. Cruise missiles are rocket powered only in the initial phase of flight. This is also wrong. Agni 5 is a medium range supersonic cruise missile. Agni 5 is a long range hypersonic ballistic missile, intercontinental ballistic missile. BrahMos is a solid fuel intercontinental ballistic missile. Not at all. It is supersonic cruise missile. So both statement 1 and statement 2 are wrong. Correct answer is neither 1 nor 2. And then there was a question, I have included this in defense, but I could have added also in space. Radar imaging. Radar imaging has applications in many areas. In the last class I was talking about remote sensing techniques. And I was saying that how remote sensing can occur with help of visible light, but can also occur with help of radio wave. And the benefit of radio waves is that they can cross through many solid dense objects. Just say if you are using radar imaging with help of planes or satellites, they can easily cross through cloud cover. The question is that with help of radar imaging, in how many of the above activities radars are being used? Identification of narcotics on passengers at airports or in the aircraft, monitoring of precipitation, tracking the migration of animal. Many institutes have given this answer as D. I don't think D will be the answer. Sorry, C. They have given all 3 to be correct. I don't think first is correct. अब UPSC का ऑफिशियल आंसर किया आएगा तो हम देखेंगे इसका क्या आंसर है, बट मेरे हिसाब से इसका आंसर ओनली 2 होना चाहिए. B should be correct. Second and third are well established applications. These are cloud patterns. We can use जो हम IMD के जो devices हैं, Indian Meteorological Department जब weather prediction करती है, then they measure parameter based on many technologies. They have weather balloons, sensors. One of that is Doppler radar. So radar looks at the cloud pattern, the moisture in the cloud, etc., etc. And based on that we do weather forecast. तो ये जो part है monitoring of precipitation, that is very simple. We can do that by monitoring cloud. This is also their tracking of migration of animals. This is actually packets of animals which are migrating. ये जो this was Doppler radar, knowing about a cloud system. ये जो bright part दिख रहा है. So radar imaging cannot tell you about one individual animal, but a group of herd of animal or birds that can be tracked, and all of these bright images which you see, these are this group of animals which are going from one place to another place. So how a large herd migrates across planet Earth, let's say in a very huge grassland area, कि tropical savanna में migration pattern क्या है, right? हमको बिना locally घूमते हुए हमको दूर से देखना है, we can do radar imaging for that. So this is also true. But that first statement, why I do not think this to be true? First of all, let me tell you that radio imaging is actually done at airports, but then calling it radar does not make sense. Radar का purpose होता है that it sends a signal and then it measures how much time it took for that signal to be reflected, and based on that it creates a map. This is more about imaging with help of radio wave. Some airports are equipped with this. तो पहले तो मैं word radar भी use करूँ वो भी प्रॉब्लमेटिक है यहाँ पे, बट चलो फिर भी मैं रेडियो इमेजिंग को मैं रडार वर्ड यूज कर भी लूँ. Even then, look at this statement carefully. That identification of narcotics on passengers at airports or in the aircraft. That in the aircraft I will never accept. That this radio radar imaging, whatever happens at the airport, it occurs with help of such gates. And at these gates, we can know if some person is hiding something. Radio waves can cross through solid objects. But saying that it is also, we can do the same thing also inside the plane, I will not accept that. And I will repeat, मेरा उससे भी पहले जो मेरा जो reservation है वो ये है that not every radio imaging should be called radar, use of radar. And even if I am doing that, then that in the airport, I will not accept it. So according to me, the answer should be only 2. B should be the correct answer. And one more question of defense. This for me was a very simple question. How many of the above are considered 5th generation fighter aircraft? India does not have 5th generation fighter aircraft. You only need to know this information. हमारे पास जो सबसे advanced fighter jet है वो Rafale है. And Rafale belongs to a generation called 4.5. जब हम generation word use करते हैं, it means that things are improving. First generation planes were made during World War II. Second generation in 60s, third generation 80s, fourth generation in 2000. Few planes which belong to America are called 5th generation fighter jet. Much more capable in flight, in maneuverability, in their stealth capability. They're much more capable. But India does not have any of the fighter planes, which is 5th generation fighter plane. If you knew this, and the सबसे advanced जो Rafale है, which we bought from France, this also belongs to generation 4.5. If you know this, then you know the answer will be none of the above. This is MiG, that is Tejas. These two are small fighter jet. Rafale is very large fighter jet, but none of them are 5th generation. Answer here is none. D is the correct answer. Let's briefly, few more questions are left. Let's discuss them. जल्दी से इसको खत्म करते हैं. International Thermonuclear Experimental Reactor. This is a magnetic fusion device under construction facility in France. India is a member along with many countries. जब ये successful हो जाएगा, then we have figured out how to produce energy using nuclear fusion. In nuclear fusion, you take deuterium, tritium together and you make helium and you produce energy. तो यहाँ पे 4 ऑप्शन में इकलौता ऑप्शन जहाँ पे फ्यूजन लिखा भी हुआ है वो डी ऑप्शन है. क्वेश्चन क्या है? that India is member of ITR. If it succeeds, what is the immediate advantage? The answer is it can build fusion reactors for power generation. Correct answer is D here. I do expect this topic to be important in subsequent examinations also. Fusion in general, right? Magnetic fusion, inertial fusion, ITR in particular. IAEA safeguards कुछ न्यूक्लियर रिएक्टर्स पे हैं, कुछ पे नहीं हैं. जब हमने पोखरन टेस्ट किया था, then lot of sanctions were imposed upon India. इंडिया ने चुपके परमाणु बम बना लिया. So we adopted lot of transparency measures that we are a responsible nuclear power. जैसे countries जो हैं, they stopped supplying uranium to India. India is not a member of nuclear supplier group. So India said to IAEA, which is an agency of United Nations, that if India does receive uranium from other countries, and if we are using that uranium in any of our reactor, then you can come and see for what purpose we are using that uranium. अगर आपका ये डर है कि इंडिया को यूरेनियम दो और बॉम्ब बनाना शुरू कर दे, देन आप आके देख सकते हो फॉर व्हाट पर्पस वी आर यूजिंग. बट जो हमारा डोमेस्टिक यूरेनियम प्रोडक्शन है वो तो हमारा प्रेरोगेटिव है, वो हम जैसे भी डिसाइड करें वो हमारा मन है. सो यहाँ पे जो आंसर है वो सिंपल है, बी इज द करेक्ट आंसर. सम यूज इंपोर्टेड यूरेनियम, अदर्स यूज domestic supply. This was to ensure that we are a responsible nuclear powered country, right? If we are receiving uranium from other countries, then it will be used for energy production in nuclear reactor, not for a nuclear bomb. This is another question, although it can be discussed in economy also. India, despite having uranium deposits, depends on coal for most of its electricity production. That is true. Uranium enriched to extent of at least 60% is required for the production of electricity. That is not at all true. Enrichment of uranium का मतलब होता है जो यूरेनियम नेचुरली मिलता है हमको नेचर में, वो यूरेनियम 235 है, यूरेनियम 238 है. 238 इज नॉट गुड एट फिशन, 235 इज. बट 235 का जो परसेंटेज है वो 0.7% है. अगर आपने 100 ग्राम यूरेनियम आपके पास है तो आपके पास 1 ग्राम से कम उसमें यूरेनियम 235 है. 99 ग्राम से ज़्यादा आपके पास यूरेनियम 238 है, विच इज़ नॉट यूजफुल फॉर फिशन. सो इनरिचमेंट में हमारा पर्पस होगा that somehow we can remove uranium 238 so that the percentage of 235 keeps on increasing. 3-4% is more than enough for nuclear reactor. 90% is very good enrichment for atomic bomb. Right? So uranium enrichment of low enrichment, which is 3-4-5%, is used in case of nuclear reactor. Statement 2 is wrong, statement 1 is correct. Answer is C here. There was a question this year on radioisotope thermoelectric generator. Like I have been talking about this topic for quite some time. I was thinking of ignoring this topic now because it was in news many years ago. But then UPSC asked a question in 2024. बहुत सारे जो outer space में missions हैं, which are very far away from Earth, very far away from Sun, they cannot rely upon solar energy. राइट, सोलर एनर्जी आउटपुट जो है सेम नहीं है और आप जितने दूर चले जाओ वो आउटपुट आपका कम होते चले जाएगा. नासा ने बहुत सारे मिशंस लॉन्च कर रखे हैं, जुपिटर, सैटर्न, काइपर बेल्ट, वॉयजर 1, वॉयजर 2 तो सोलर सिस्टम का भी पार्ट नहीं है, बट स्टिल इट्स सेंडिंग सिग्नल. सो हाउ आर दे बीइंग पावर्ड? कुछ तो एनर्जी चाहिए ना. सो दे आर बीइंग पावर्ड विथ हेल्प ऑफ दैट इनसाइड दिस स्पेसक्राफ्ट यू विल फाइंड देयर इज अ कंटेनर, एंड इन दिस कंटेनर वी कीप अ रेडियो आइसोटोप plutonium 238. And that plutonium 238 is a radioactive material which emits alpha particle. It decays into uranium, but it also releases energy. And that energy is thermal energy. But ये जो wall जिस material का बना है, it can convert that thermal energy into electrical energy, and it is connected to a battery and it can charge a battery. तो इसका benefit ये है that solar energy पे मेरा reliance का कोई ज़रूरत नहीं है. I have plutonium inside it. It's not fission. It should be very clear that ये fission नहीं है, ये radioactivity है. That plutonium is releasing an alpha particle, which essentially is helium. Plutonium has atomic, there are many isotopes, we use this isotope. When it releases this helium, helium is nothing but 2He4. So its atomic number is 92, then it becomes uranium and it becomes uranium 234. But it releases energy in this reaction. And that energy is absorbed by the wall as thermal energy, which gets converted into electrical energy. And that is how you power your spacecraft such as Voyager 1 or some rover on the surface of the Mars. Very popular technology used by NASA for many years now. Why this topic was in news, that ISRO is exploring this technology, that we should also try to use this technology for future space exploration. And this was, I did find a reference to this technology in a news from July 2023 in one of the Hindu article. So RTGR miniature fission reactor, बिल्कुल ही गलत है, ये fission reaction नहीं है, ये radioactivity है. If I know first to be wrong, I know my answer is 2 and 3. But look at the second option. RTGs are used for powering the onboard system of spacecraft. That is true. RTG can use plutonium 238, which is a byproduct of weapon development. Okay, that statement, even though you do not know, your answer is not dependent upon it. But remember, uranium nature में बहुत ज़्यादा है. Uranium की nature में कोई कमी नहीं है. But plutonium की बहुत कमी है. और plutonium particularly 238 की बहुत ही ज़्यादा कमी है. जो plutonium 238 है, when you enrich uranium for weapons, then plutonium 238 is one of the byproduct which is used in space technology also. So that statement is also true. So answer here is 2 and 3 only. Last few questions. Fuel cell. Fuel cell is this hydrogen based cell where at the cathode and at the anode you have hydrogen and oxygen, and you have hydrogen is being converted into proton, which is allowed to pass through membrane, but electrons are not allowed. Electrons are forced to go through some external circuit, and that is how you gain energy. And then it mixes with oxygen. So now you have proton, electron, oxygen, and they make H2O, water vapor. If pure hydrogen is used as a fuel, the fuel cell emits heat and water as byproduct. ये यूपीएससी ने 2024 में भी सेम चीज पूछा है, that fuel cell का byproduct क्या होगा? The first is true. Fuel cells can be used for powering buildings and not for small devices. ये बिल्कुल गलत है. I can have fuel cell powering ships and trains, but also mobile phones and laptops as long as I have some source of hydrogen. The only thing is that fuel cell is not like a battery, because battery को आप charge discharge कर सकते हो. फ्यूल सेल में हमेशा आपको हाइड्रोजन कैरी करके रखना पड़ेगा. यू नीड टू हैव अ मैकेनिज्म वेयर सम हाइड्रोजन इज अवेलेबल टू यू. फ्यूल सेल्स प्रोड्यूस इलेक्ट्रिसिटी इन फॉर्म ऑफ AC. As a thumb rule, remember that if it is a battery, if it is a cell, it will produce DC. If it is some sort of rotating device such as turbine, it will produce AC. So wind turbine, in general turbines will produce AC. Battery cells, जैसे photovoltaic cell जो है, solar panel है, that will produce DC. Fuel cell will produce DC. It goes without saying that we can always convert AC to DC and DC to AC. There are devices capable of doing that. तो यहाँ पे जो आंसर है वो one only है. This is prima facie, it looks like a very difficult question, but ये one of the simplest question है. That solar power can be used for running surface pumps, not for submersible pump. ऐसा क्यों होगा? If it is power, then if submersible pumps are being used by some energy source, I can produce with help of solar energy. Solar power can be used for running centrifugal pumps, not ones with piston. Again, ऐसा क्यों होगा? I can always convert one form of energy into another form of energy. अगर आप मान लो एक सिंपल सा एग्जांपल ले लेते हैं कि मान लो हम पैनल नहीं यूज़ कर पा रहे हैं, मिरर यूज़ कर लेंगे. मिरर को हम कॉन्सेंट्रेट करेंगे सनलाइट को पानी बॉयल करने के लिए. दैट वाटर एज इट बॉइल्स अप, it produces steam. Let's connect that steam to a turbine and produce electricity. So ऐसा क्यों होगा कि why we will not be able to do one? So prima facie it looks like a very difficult question, but it is not. It's a very simple question based on understanding. Answer here is neither one nor two. Both of these statements are wrong here. Green hydrogen, I will leave this question for environment discussion. Only one question of IPR. According to Indian Patent Act, a biological process to create a seed can be patented in India. That is not true. You cannot do that. In India, there is no intellectual property appellate board. ये देखो कैसा statement है. That अगर नहीं है तो हमने एक made up किया है एक पूरा एजेंसी और हम बोल रहे हैं कि ये एजेंसी नहीं है. तो ये बड़ा अजीब सा लग रहा है सोच के कि हमने एक made up एजेंसी, अगर मान लो ये स्टेटमेंट गलत है तो इसका मतलब ये है कि पहले तो हमने एक मन में बनाया एक एजेंसी और हम पूछ रहे हैं कि देखो ये एजेंसी नहीं है इंडिया में. दैट डज नॉट लुक गुड. सो एनीवेज देयर इज वन, ऑल्दो इट वाज रिसेंटली डिसबैंडेड. सो एज ऑफ नाउ दैट स्टेटमेंट इज ट्रू, बट एट दैट पॉइंट ऑफ टाइम दैट स्टेटमेंट इज नॉट ट्रू. थर्ड इज प्लांट वेराइटीज आर नॉट एलिजिबल टू बी पेटेंटेड इन इंडिया. दैट इज ट्रू. पेटेंट लॉ आप जाओगे, लिविंग ऑर्गेनिजम्स कैन नॉट बी पेटेंटेड. एंड दिस लास्ट टॉपिक, some of the topics of material sciences and nanotechnology. With reference to use of nanotechnology in health sector, targeted drug delivery is made possible by nanotechnology. ये सबसे prominent application है that हम specific cells and tissues को target कर सकते हैं rather than consuming a medicine which is not targeting specific cells and tissues. Can largely contribute to gene therapy. Remember, जब भी हमको gene therapy करना हो, we need very precise changes to inside our cell, and this is where nano carriers come into place. So even this part is true. Nanotechnology का एक जो सबसे बड़ा application biology में इसीलिए है that most of the biological processes occur at nano level. आपका जो DNA है वो 2 नैनोमीटर डायमीटर का मॉलिक्यूल है. उसका लेंथ बहुत ज़्यादा है, बट उसका जो डायमीटर है 2 नैनोमीटर का है. जो आपका हीमोग्लोबिन है वो 5 नैनोमीटर डायमीटर का है. So your most fundamental biological process inside body occurs at the level of nano. So if you want to make some changes at such scale, then you'll have to use nano-engineered materials. So answer is both 1 and 2. 3D printing, this layer by layer manufacturing, all of them are correct. Preparation of confectionery item, bionic ears, automotive industry, reconstructive surgery, data processing technology, सब में application है. 3D printing has come a long way. It started with making small toys of plastic layer by layer, but now we are using it to make chips and metallic objects and buildings also. There are 3D printed houses now these days. which are becoming popular. पूरा house जो है 2-3 दिन में बन सकता है, which can take few weeks to be constructed. Carbon nanotube, they can be used as a carrier of drugs and antigens in the human body. Carbon nanotube is this cylindrical tube folded out of graphene sheet, and graphene is this 2D arrangement of carbon atoms, a two-dimensional arrangement of carbon atom. Graphene in itself is quite amazing, and carbon nanotube becomes even more amazing depending upon how you have folded it. Carbon nanotube is a nanomaterial जिसका जो cylindrical tube का जो diameter होगा वो कुछ नैनोमीटर का होगा. अब उस cylindrical tube के अंदर you can carry some other nanomaterial, and carbon nanotube isolates this nanomaterial with the surrounding. So carbon nanotube can act as a carrier, and you can, जो अभी हमने discuss किया था targeted drug delivery, that can occur with help of carbon nanotube. And even for vaccine, I can use this, hence the word antigens. They can be made into artificial blood capillaries for an injured part of human body. They were explored at one point of time. That carbon nanotubes are biodegradable. मान लो sudden blood loss है, इससे पहले कि आप सर्जरी वगैरह जो भी करो, if you have to make artificial capillaries कि blood loss कम से कम बंद हो, then you have to ensure that whatever you are using should not produce any toxicity inside body. Because फिर इंसान blood loss से पहले toxicity से मर जाएगा. So exploring those things, that is one of the biggest challenge, right? Metallic objects clearly cannot be used. One of the reason why carbon nanotubes were explored, because many of them are biodegradable, right? But nonetheless, I don't think they actually are being used anywhere. In any case, second statement is not correct. Third is correct. It is a nanomaterial, can be used as a sensor to know about some parameter, atmospheric parameter, some chemicals, etc. So third is true. And fourth is also true, that one of the reason they were being explored was this, that they are biodegradable. But second cannot be considered true, that हम इसको actually में use कर रहे हैं. Although statement यहाँ पे can था, थोड़ा controversial statement है. And I will repeat that they were actually explored for this purpose. But that result, that did not lead to anything concrete, that they can be made into some artificial blood capillaries. So यहाँ पे जो answer है वो 1, 3 and 4 है. And given the fact that there is this option 1, 2, 3 and 4, this becomes a difficult question, right? That you may think that even that second statement is true. Bisphenol A, that is used in polycarbonate. Polycarbonate जो है thick, जो strong plastics जो होते हैं, not the plastic polythene bag I'm talking about. Think of those thick, strong plastic. In that we use this bisphenol A, and bisphenol A helps in maintaining that structure. The problem is that in some food cans, if you are using bisphenol A, if it is leaking in your food item, then it is a toxic substance. So it has been considered a cause of concern, and because of that it remains in news a lot. And triclosan, I feel like that these are lazy questions. किसी को question कम पढ़ रहे थे तो जो एक दो उन्होंने देखा, because एक ही साल का question है, ये भी 2021 का question है, और आसपास आपने दो चीज़ों बस पूछ ले कि कौन पर्पस के लिए आप यूज़ करते हो. So जो triclosan है, this is used as, triclosan essentially is this antiseptic material, antibacterial material, that it does not allow growth of bacteria and other microbes. And in toiletries, जो डे टू डे हमारा बॉडी केयर है, साबुन में, टूथपेस्ट में, इन चीज़ों में इसका यूज़ है. बट प्रॉब्लम ये है कि बियॉन्ड अ सर्टेन वैल्यू, इफ़ दे आर अगेन बीइंग कंज्यूम्ड बाय बॉडी, it can lead to various type of diseases, and because of that it remains in use. So answer यहाँ पे D है, that triclosan is used in toiletries. Another question on nanotech. Other than made by humans, nanoparticles do not exist in nature. ये बिल्कुल ही गलत है. जो pollens होते हैं वो nanoparticles हैं. बहुत सारे जो pollutants हैं, aerosols हैं, वो आपके nanoparticles हैं. जैसे हम बात करते हैं ना particulate matter 2.5. That 2.5 is 2.5 micrometer, but वो कम होते होते वो 50-100 नैनोमीटर भी हो सकता है वो. So ये बिल्कुल ही गलत है. Volcanic eruption के जो particles होंगे, many of them can be nanoparticle. Nanoparticles of some metallic oxide are used in manufacture of some cosmetic. I will give you two best example. Titanium dioxide, zinc oxide used in sunscreen, and a nanomaterial of called liposome which is used in lipsticks. So second statement is also true. Second statement is true. First is not correct. Nanoparticles which enter the environment are unsafe for humans, some of them. That is one of the biggest concern of nanotechnology, that some of the nanomaterials can be danger to human health. So 2 and 3 are correct. First is not correct. Answer here is D. तो ऑप्शन भी यूपीएससी ने थोड़ा बढ़िया अरेंज करके दिया था कि कम से कम देखो एक तो गलत है, कि आउट ऑफ द 3 स्टेटमेंट्स एटलीस्ट 1 इज रॉन्ग एंड एटलीस्ट 1 इज करेक्ट. अगेन सेम थिंग. So I need to find at least 1 correct and at least 1 wrong. If you can think it like this, you will find the right answer even if you did not know the exact information. A question on again material science. Carbon fibers are used in manufacture of components used in automobile. Carbon fiber looks like this, made of carbon layer, very strong material, and this reduces mass of heavy objects without compromising on the strength of those things. And because of that, it is being used in making vehicles, even planes also. So that first part is actually true, that carbon fibers are used in manufacture of components used in automobiles and aircrafts. Once used cannot be recycled. This does not seem at all correct. अगर ऐसा भी स्टेटमेंट रहता कि इसका रिसाइकलिंग करना मुश्किल है, तो फिर भी मैं सोचता एक सेकंड के लिए कि क्या पता है स्टेटमेंट सही है. बाय द वे ये सही है, कार्बन फाइबर को रिसाइकल करना ज़्यादा मुश्किल है. बट ये कहना कि दे कैनॉट बी रिसाइकल्ड एट ऑल, दैट डज नॉट मेक सेंस. तो ऐसे जब भी एक्सट्रीम स्टेटमेंट्स हो, डेफिनेटली कंसिडर देम रॉन्ग. जहाँ पे भी ऐसे लगे कि आपने एक ब्लैंकेट स्टेटमेंट दे दिया, राइट, कि ये हो ही नहीं सकता, मोस्ट ऑफ द टाइम जो है वो गलत होगा. आंसर हियर इज 1 ओनली. एंड द लास्ट क्वेश्चन ऑफ दिस एंटायर डिस्कशन, hydrogel pe there was a question. Hydrogel is this very amazing material. Some of them are found in nature also. Many of them are made into industrial setup. Hydrogels are known for extremely hydrophilic molecules. Hydrophilic molecules attract water. Hydrophobic molecules repel water. So hydrogel, even though it looks like this jelly solid object, हाइड्रोजेल कैन हैव एज लार्ज एज 90% वाटर. इसमें जो 90% है सिर्फ पानी है. अनदर वेरी इंटरेस्टिंग मटेरियल इज एरोजेल, जहाँ पे 90-95% एयर होगा, दिखेगा सॉलिड बट अंदर पूरा एयर भरा पड़ा है. उसके अपने खुद के अलग सेट ऑफ एप्लीकेशंस हैं. द बेस्ट पार्ट ऑफ हाइड्रोजेल इज कि कितना पानी आपको रिलीज करना है, कितना नहीं करना है. दिस यू कैन कंट्रोल विथ हेल्प ऑफ कंट्रोलिंग टेम्परेचर, pH, मल्टीपल थिंग्स. सो इसका जो एक सबसे फेमस एप्लीकेशन है वो कंट्रोल ड्रग डिलीवरी में है. दैट हाइड्रोजेल यूजिंग सम थेराप्यूटिक मेडिसिन, बट यू डू नॉट वांट दैट मेडिसिन टू बी रिलीज्ड इन द ब्लड स्ट्रीम इन वन गो. यू वांट टू बी रिलीज्ड इट ग्रेजुअली, राइट. तो बॉडी में pH वैल्यू जैसे चेंज होगा वैसे वैसे रिलीज होगा. सो हाइड्रोजेल का फायदा ये है दैट इट इज, तो हम क्या करेंगे, ये जो पानी है, दैट मेडिसिन शुड बी डिसॉल्वेबल इन वाटर. सो नाउ इट इज नो लॉन्गर जस्ट वाटर. That whatever that medicine is, it has been dissolved in water, and this hydrogel carries that therapeutic medicine, and based on the parameters as they change, it can release that medicine into body. Mobile air conditioning system, one of the amazing application. Remember, water when it when it vaporizes, it leads to cooling condition. तो एक सबसे इंटरेस्टिंग तरीका जो है, बहुत सारा पानी, हाइड्रोजेल का सरफेस एरिया बहुत ज़्यादा हो सकता है, बहुत सारा पानी अगर आपको कैरी करना हो, सो दैट इट कैन हेल्प इन वेपराइजेशन. इन दिस मोबाइल एयर कंडीशनिंग सिस्टम, दिस इज बीइंग एक्सप्लोर्ड. Preparation of industrial lubricants, the same thing. Many of the hydrogels are also being used in reducing friction in industrial lubricant. Answer here is all 3 of them. So correct answer is 1, 2 and 3. This concludes our discussion about all of the questions of science and tech from last 10 years. And if you have any questions, if you want to ask about any topic more, always send me a mail. And you can download this PPT and the PDF which I was talking about in the last class from the video when it will be uploaded on YouTube. All the best, guys.